The glycogen storage diseases result from a hereditary deficiency of one of the enzymes involved in the synthesis or sequential degradation of glycogen. Depending on the normal tissue or organ distribution of the specific enzyme, glycogen storage in these disorders may be limited to a few tissues, may be more widespread while not affecting all tissues, or may be systemic. The significance of a specific enzyme deficiency is best understood from the perspective of the normal metabolism of glycogen. Figure 5.14. Glycogen is a storage form of glucose. Glycogen synthesis begins with the conversion of glucose to glucose-6-phosphate by the action of a hexokinase glucokinase. A phosphoglucomutase then transforms the glucose-6-phosphate to glucose-1-phosphate, which in turn is converted to uridine diphosphoglucose. A highly branched, large polymer is then built molecular weight as high as 100 million, containing as many as 10,000 glucose molecules linked together by alpha-1,4-glucoside bonds. The glycogen Mendelian disorders 161. Chain and branches continue to be elongated by the addition of glucose molecules mediated by glycogen synthetases. During degradation, distinct phosphorylases in the liver and muscle split glucose-1-phosphate from the glycogen until about 4 glucose residues remain on each branch, leaving a branched oligosaccharide called limit dextrin. This can be further degraded only by the debranching enzyme. In addition to these major pathways, glycogen is also degraded in the lysosomes by acid alpha-glucosidase. If the lysosomes are deficient in this enzyme, the glycogen contained within them is not accessible to degradation by cytoplasmic enzymes such as phosphorylases. On the basis of specific enzyme deficiencies and the resultant clinical pictures, glycogenoses have traditionally been divided into a dozen or so syndromes designated by 7 liver phosphorylase, kinase, pi, glycogen lysosomal degradation 1 activates, phosphorylase V, Vi, branching enzyme Phi, I, glycogen synthetase. Limit dextrin 4 glucose residues in branch configuration, glucose-1-phosphate. Uridine diphosphoglucose I, lysosomal, PP acid maltases 2. UTP-I. Debranching enzyme 2 phosphoglucomutase. Glucose-6-phosphate. Glucokinase glucose-PI glucose-6-phosphatase I, phosphoglucoisomerase. Fructose-6-phosphate. Phosphofructokinase is 7th. Fructose-1, 6-diphosphate. Pyruvate lactate. Figure 5.1. 4 pathways of glycogen metabolism. Asterisks mark the enzyme deficiencies associated with glycogen storage diseases. Roman numerals indicate the type of glycogen storage disease associated with the given enzyme deficiency. Types V and VI result from deficiencies of muscle and liver phosphorylases, respectively. Modified from Hers, H. et al. Glycogen storage diseases. Inscriver C.R. et al. Editors. The Metabolic Basis of Inherited Disease, ed. 6, New York, 1989, McGraw-Hill, p. 425. 162nd, Chapter 5, Genetic Disorders. Clinical pathologic category. Hepatic type, myopathic type, miscellaneous types. Subgroups of glycogeos. 5 in principle. ES7, enzyme. Specific type deficiency. Hepatorenal, von glucose. 6 Gierke disease phosphatase type 1, McArdle disease. Type V, muscle phosphorylase, lysosomal acid alpha-glucosidase. Generalized glycogenosis, Pompe disease type 2. Morphologic changes, hepatomegaly, intracytoplasmic accumulations of glycogen and small amounts of lipid, intranuclear glycogen rhinomegaly. Intracytoplasmic accumulations of glycogen in cortical tubular epithelial cells. Skeletal muscle only, accumulations of glycogen predominant in subsarcolemmal location. Mild hepatomegaly, ballooning of lysosomes with glycogen, creating lacy cytoplasmic pattern cardiomegaly, glycogen within sarcoplasm as well as membrane-bound skeletal muscle, similar to changes in heart. Clinical features in untreated patients, failure to thrive, stunted growth, hepatomegaly, and renomegaly, hypoglycemia due to failure of glucose mobilization, often leading to convulsions, hyperlipidemia, and hyperuricemia resulting from deranged glucose metabolism. Many patients develop gout and skin xanthomas bleeding tendency due to platelet dysfunction with treatment. Most survive and develop late complications, e.g. hepatic adenomas. Painful cramps associated with strenuous exercise. Myoglobinuria occurs in 50% of cases. Onset in adulthood greater than 20 years. Muscular exercise fails to raise lactate level in venous blood. Serum creatine kinase always elevated, compatible with normal longevity. Massive cardiomegaly, muscle hypotonia, and cardiorespiratory failure within 2 years, a milder adult form with only skeletal muscle involvement, presenting with chronic myopathy, enzyme replacement therapy available. Roman numerals, and the list continues to grow. On the basis of pathophysiology, glycogenosis can be divided into 3 major subgroups, Table 5.7. Hepatic forms. The liver is a key player in glycogen metabolism. It contains enzymes that synthesize glycogen for storage and ultimately break it down into free glucose, which is then released into the blood. An inherited deficiency of hepatic enzymes that are involved in glycogen degradation therefore leads not only to the accumulation of glycogen in the liver, but also to a reduction in blood glucose concentrations, hypoglycemia. Figure 5.15. Deficiency of the enzyme glucose 6-phosphatase von Gierke disease, or type 1 glycogenosis, is a prime example of the hepatic hypoglycemic form of glycogen storage disease. See Table 5.7. Other examples include deficiencies of liver phosphorylase and debranching enzyme, both involved in the breakdown of glycogen. See Figure 5.15. In all these disorders, glycogen is stored in many organs, but hepatic enlargement and hypoglycemia dominate the clinical picture. Myopathic forms. In the skeletal muscles, as opposed to the liver, glycogen is used predominantly as a source of energy during physical activity. Adenosine triphosphate ATP is generated by glycolysis, which leads ultimately to the formation of lactate, Figure 5.16. If the enzymes that fuel the glycolytic pathway are deficient, glycogen storage occurs in the muscles and is associated with muscular weakness due to impaired energy production. Examples in this category include deficiencies of muscle phosphorylase myocardial disease, or type V glycogenosis, muscle phosphofructokinase, type 7 glycogen storage disease, and several others. Typically, individuals with the myopathic forms present with muscle cramps after exercise and lactate levels in the blood fail to rise after exercise due to a block in glycolysis. Glycogen storage disease is associated with 1 deficiency of acid alpha glucosidase acid maltase and 2 lack of branching enzyme do not fit into the hepatic or myopathic categories. They are associated with glycogen storage in many organs and death early in life. Acid alpha glucosidase is a lysosomal enzyme, and hence its deficiency leads to lysosomal storage of glycogen type 2 glycogenosis, or Pompe disease, in all organs, but cardiomegaly is the most prominent feature. See Figure 5.16. Key concepts: glycogen storage diseases. Inherited deficiency of enzymes involved in glycogen metabolism can result in storage of normal or abnormal forms of glycogen, predominantly in liver or muscles, but also in other tissues as well. In the most common hepatic form, von Gierke disease, liver cells store glycogen because of a lack of hepatic glucose 6-phosphatase. The liver is enlarged and patients have hypoglycemia. There are several myopathic forms, including myocardial disease, in which lack of muscle phosphorylase gives rise to storage in skeletal muscles and cramps after exercise. In Pompe disease, there is lack of lysosomal acid alpha glucosidase, and all organs are affected, but heart involvement is predominant. Table. Multigenic disorders 163. A. Liver, glycogen, glucose, muscle, normal, blood glucose, glycogen, glycogen storage disease, hepatic type, various tissues, glycolysis, energy, disorders associated with defects in proteins that regulate cell growth. Normal growth and differentiation of cells are regulated by 2 classes of genes, proto-oncogenes and tumor suppressor genes, whose products promote or restrain cell growth. Chapter 7. It is well established that mutations in these 2 classes of genes are important in the pathogenesis of tumors. In the vast majority of cases, cancer-causing mutations affect somatic cells and hence are not passed in the germline. In approximately 5% of all cancers, however, mutations transmitted through the germline contribute to the development of cancer. Most familial cancers are inherited in an autosomal dominant fashion, but a few recessive disorders have also been described. This subject is discussed in Chapter 7. Specific forms of familial tumors are described in various chapters. BC, glycogen glucose, glycogen storage disease, myopathic type, glycolysis, glucose, low energy output, glycogen, Figure 5.15a, normal glycogen metabolism in the liver and skeletal muscles. B. Effects of an inherited deficiency of hepatic enzymes involved in glycogen metabolism. C. Consequences of a genetic deficiency in the enzymes that metabolize glycogen in skeletal muscles. Low blood glucose, Complex multigenic disorders. As discussed previously, such disorders are caused by interactions between variant forms of genes and environmental factors. A gene that has at least two alleles, each of which occurs at a frequency of at least 1% in the population, is polymorphic, and each variant allele is referred to as a polymorphism. According to the common disease, common variant hypothesis, complex genetic disorders occur when many polymorphisms, each with a modest effect and low penetrance, are co-inherited. Two additional facts have emerged from studies of common complex disorders, such as type 1 diabetes. While complex disorders result from the collective inheritance of many polymorphisms, different polymorphisms vary in significance. For example, of the 20 to 30 genes implicated in type 1 diabetes, 6 or 7 are most important, and a few HLA alleles contribute more than 50% of the risk. Chapter 24. A B figure 5.1. 6 pump disease, glycogen storage disease type 2. A normal myocardium with abundant eosinophilic cytoplasm. B patient with Pompe disease, same magnification, showing the myocardial fibers full of glycogen seen as clear spaces. Courtesy Dr. Trace Worrell, Department of Pathology, University of Texas Southwestern Medical Center, Dallas, Tex. Complex. Chapter 5. Genetic disorders. Some polymorphisms are common to multiple diseases of the same type, while others are disease-specific. This is best illustrated in autoimmune diseases. Chapter 6. Several normal phenotypic characteristics are governed by multifactorial inheritance, such as hair color, eye color, skin color, height, and intelligence. These characteristics show a continuous variation in population groups, producing the standard bell-shaped curve of distribution. Environmental influences, however, significantly modify the phenotypic expression of complex traits. For example, type 2 diabetes has many of the features of a multifactorial disorder. It is well recognized that individuals often first manifest this disease after weight gain. Thus, obesity, as well as other environmental influences, unmask the diabetic genetic trait. Nutritional influences may cause even monozygous twins to achieve different heights. A culturally deprived child cannot achieve his or her full intellectual capacity. Assigning a disease to this mode of inheritance must be done with caution. It depends on many factors, but first on familial clustering and the exclusion of Mendelian and chromosomal modes of transmission. A range of levels of severity of a disease is suggestive of a complex multigenic disorder, but, as pointed out earlier, variable expressivity and reduced penetrance of single mutant genes may also account for this phenomenon. Because of these problems, sometimes it is difficult to distinguish between Mendelian and multifactorial disease. Chromosomal disorders. Normal karyotype, as you will remember. Human somatic cells contain 46 chromosomes, 22 homologous pairs of autosomes, and 2 sex chromosomes, XX in the female and XY in the male. The study of chromosomes, karyotyping, is the basic tool of the cytogeneticist. The usual procedure to examine chromosomes is to arrest dividing cells in metaphase with mitotic spindle inhibitors, e.g., N-diacetyl N-methylcolchicine, cosemid, and then to stain the chromosomes. In a metaphase spread, the individual chromosomes take the form of 2 chromatids connected at the centromere. A karyotype is obtained by arranging each pair of autosomes according to length, followed by sex chromosomes. A variety of staining methods have been developed that allow identification of individual chromosomes on the basis of distinctive and reliable patterns of alternating light and dark bands. The one most commonly used involves a Giemsa stain and is hence called G-banding. A normal male karyotype with G-banding is illustrated in figure 5.17. With standard G-banding, approximately 400 to 800 bands per haploid set can be detected. The resolution obtained by banding can be markedly improved by obtaining the cells in prophase. The individual chromosomes appear markedly elongated, and as many as 1,500 bands per karyotype can be recognized. The use of these banding techniques permits certain identification. Arm region band subband, PQ, 2, 1, 1, 2, 2, 1, 1, 2, 3, 1, 2, 3, 4, 5, 6, 3, 2, 1, 3, 2, 1, 4, 3, 6, 1, 1, 2, 1, 2, 3, 1, 2, 3, 1, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, XY, 2, 3, 4, 5, X chromosome. Figure. G banded karyotype from a normal male, 46XY, also. Shown is the banding pattern of the X chromosome with nomenclature of arms, regions, bands, and subbands. Courtesy Dr. Stuart Schwartz, Department of Pathology, University of Chicago, Chicago, Ill. 165. Chromosomal disorders 165 of each chromosome and roughly delineates breakpoints and other gross alterations described later. Commonly used cytogenetic terminology karyotypes are usually described using a shorthand system of notations in the following order. Total number of chromosomes is given first, followed by the sex chromosome complement, and finally the description of abnormalities in ascending numerical order. For example, a male with trisomy 21 is designated 47XY21. Notations denoting structural alterations of chromosomes and their corresponding abnormalities are described later. The short arm of a chromosome is designated P for petite, and the long arm is referred to as Q, the next letter of the alphabet. In a banded karyotype, each arm of the chromosome is divided into 2 or more regions bordered by prominent bands. The regions are numbered e.g. 1, 2, 3 from the centromere outward. Each region is further subdivided into bands and subbands, and these are ordered numerically as well. See figure 517. Thus, the notation XP212 refers to a chromosomal segment located on the short arm of the X chromosome in region 2, band 1, and subband 2. Structural abnormalities of chromosomes. The aberrations underlying cytogenetic disorders may take the form of an abnormal number of chromosomes or alterations in the structure of one or more chromosomes. The normal chromosome complement is expressed as 46X for females and 46XY for males. Any exact multiple of the haploid number of chromosomes 23 is called euploid. If an error occurs in meiosis or mitosis and a cell acquires a chromosome complement that is not an exact multiple of 23, it is referred to as aneuploidy. The usual causes for aneuploidy are non-disjunction and anaphase lag. When non-disjunction occurs during gametogenesis, the gametes formed have either an extra chromosome N1 or one less chromosome N1. Fertilization of such gametes by normal gametes results in 2 types of zygotes, trisomic 2n1 or monosomic 2n1. In anaphase lag, one homologous chromosome in meiosis or one chromatid in mitosis lags behind and is left out of the cell nucleus. The result is one normal cell and one cell with monosomy. As discussed later, monosomies or trisomies involving the sex chromosomes, or even more bizarre aberrations, are compatible with life and are usually associated with variable degrees of phenotypic abnormalities. Monosomy involving an autosome generally causes loss of too much genetic information to permit live birth or even embryogenesis, but several autosomal trisomies do permit survival. With the exception of trisomy 21, all exhibit severe malformations and almost invariably die at an early age. Occasionally, mitotic errors in early development give rise to 2 or more populations of cells with different chromosomal complement in the same individual, a condition referred to as mosaicism. Mosaicism can result from mitotic errors during the cleavage of the fertilized ovum or in somatic cells. Mosaicism affecting the sex chromosomes is relatively common. In the division of the fertilized ovum, an error may lead to one of the daughter cells receiving 3 sex chromosomes, whereas the other receives only 1, yielding, for example, a 45x47 30th mosaic. All descendant cells derived from each of these precursors thus have either a 47X complement or a 45X complement. Such a patient is a mosaic variant of Turner syndrome, with the extent of phenotypic expression dependent on the number and distribution of the 45X cells. Autosomal mosaicism seems to be much less common than that involving the sex chromosomes. An error in an early mitotic division affecting the autosomes usually leads to a non-viable mosaic due to autosomal monosomy. Rarely, the non-viable cell population is lost during embryogenesis, yielding a viable mosaic, e.g. 46XY, 47 e.g. Such a patient is a trisomy 21 mosaic with variable expression of Down syndrome, depending on the proportion of cells containing the trisomy. A second category of chromosomal aberrations is associated with changes in the structure of chromosomes. To be visible by routine banding techniques, a fairly large amount of DNA, approximately 2 to 4 million BP, containing many genes, must be involved. The resolution is much higher with fluorescence in situ hybridization FISH, which can detect changes as small as kilobases. Structural changes in chromosomes usually result from chromosome breakage followed by loss or rearrangement of material. In the next section, the more common forms of alterations in chromosome structure and the notations used to signify them are reviewed. Deletion refers to loss of a portion of a chromosome. Figure 518. Most deletions are interstitial, but rarely terminal deletions may occur. Interstitial deletions occur when there are two breaks within a chromosome arm, followed by loss of the chromosomal material between the breaks and fusion of the broken ends. One can specify in which regions and at what bands the breaks have occurred. For example, 46XY, del 16P even 2 pu P 13 in the short arm of chromosome 16 at 16p1102, and 16p 13.1, with loss of material between breaks. Terminal deletions result from a single break in a chromosome arm, producing a fragment with no centromere, which is then lost at the next cell division. The deleted end of the retained chromosome is protected by acquiring telomeric sequences. A ring chromosome is a special form of deletion. It is produced when a break occurs at both ends of a chromosome with fusion of the damaged ends, see figure 518. If significant genetic material is lost, phenotypic abnormalities result. This might be expressed as 46XY-R14. Ring chromosomes do not behave normally in meiosis or mitosis and usually result in serious consequences. Inversion refers to a rearrangement that involves two breaks within a single chromosome with reincorporation of the inverted intervening segment, see figure 518. An inversion involving only one arm of the chromosome is known as paracentric. If the breaks are on opposite sides of the centromere, it is known as paracentric. Inversions are often fully compatible with normal development. Isochromosome formation results when one arm of a chromosome is lost and the remaining arm is duplicated, resulting in a chromosome consisting of two short arms only, or of two long arms. See figure 518. An isochromosome has morphologically identical genetic information in both arms. The most common isochromosome present in live births involves the long arm of the X chromosome and is designated IXQ10. The XQ isochromosome is associated. 166 Chapter 5 Genetic Disorders, Translocations, Balanced Reciprocal, Centric Fusion, Robertsonian Lost, Isochromosomes Deletions. Fragments, inversions, paracentric, ring chromosomes, fragments, figure 5.1, 8 types of chromosomal rearrangements, with monosomy for genes on the short arm of X and with trisomy for genes on the long arm of X. In a translocation, a segment of one chromosome is transferred to another, see figure 5.18. In one form, called balanced reciprocal translocation, there are single breaks in each of two chromosomes with exchange of material. A balanced reciprocal translocation between the long arm of chromosome 2 and the short arm of chromosome 5 would be written 46XX25Q31P14. This individual has 46 chromosomes with altered morphology of one of the chromosomes 2 and one of the chromosomes 5. Because there has been no or very little loss of genetic material, the individual is likely to be phenotypically normal. A balanced translocation carrier, however, is at increased risk for producing abnormal gametes. For example, in the case cited earlier, a gamete containing one normal chromosome 2 and a translocated chromosome 5 may be formed. Such a gamete would be unbalanced because it would not contain the normal complement of genetic material. Subsequent fertilization by a normal gamete would lead to the formation of an abnormal, unbalanced zygote, resulting in spontaneous abortion or birth of a malformed child. The other important pattern of translocation is called a Robertsonian translocation or centric fusion, a translocation between two acrocentric chromosomes. Typically, the breaks occur close to the centromeres of each chromosome. Transfer of the segments then leads to one very large chromosome and one extremely small one. Usually, the small product is lost, see figure 5.18, however, because it carries only highly redundant genes, e.g. ribosomal RNA genes. This loss is compatible with a normal phenotype. Robertsonian translocation between two chromosomes is encountered in 1 in 1,000 apparently normal individuals. The significance of this form of translocation also lies in the production of abnormal progeny, as discussed later with Down syndrome. As pointed out earlier, the clinically detected chromosome disorders represent only the tip of the iceberg. It is estimated that approximately 7.5% of all conceptions have a chromosomal abnormality, most of which are not compatible with survival or live birth. Even in live-born infants, the frequency is approximately 0.5% to 1%. It is beyond the scope of this book to discuss most of the clinically recognizable chromosomal disorders. Hence, we focus attention on those few that are most common. Cytogenetic disorders involving autosomes. Trisomy 21, Down syndrome, is the most common of the chromosomal disorders and is a major cause of intellectual disability. In the United States, the incidence in newborns is about 1 in 700. Approximately 95% of affected individuals have trisomy 21, so their chromosome count is 47. Fish with chromosome 21, specific probes reveals the extra copy of chromosome 21, in such cases, figure 519. As mentioned earlier, the most common cause of trisomy, and therefore of Down syndrome, is meiotic non-disjunction. The parents of such children have a normal karyotype, and are normal in all respects. Maternal age has a strong influence on the incidence of trisomy 21. It occurs once in 1,550 live births in women under age 20, in contrast to 1 in 25 live births for mothers. Disorders 167, figure 5.1, fluorescence in situ hybridization analysis of an interphase nucleus using locus-specific probes to chromosome 13 green and chromosome 21 red, revealing 3 red signals consistent with trisomy 21. Courtesy Dr. Stuart Schwartz, Department of Pathology, University of Chicago, Chicago, Illinois. Chromosomal. Older than age 45. The correlation with maternal age suggests that in most cases the meiotic non-disjunction of chromosome 21 occurs in the ovum. Indeed, studies in which DNA polymorphisms were used to trace the parental origin of chromosome 21 have revealed that in 95% of the cases with trisomy 21, the extra chromosome is of maternal origin. Although many hypotheses have been advanced, the reason for the increased susceptibility of the ovum to non-disjunction remains unknown. In about 4% of cases of Down syndrome, the extra chromosomal material derives from the presence of a Robertsonian translocation of the long arm of chromosome 21 to another acrocentric chromosome, e.g. 22 or 14. Because the fertilized ovum already possesses 2 normal autosomes 21, the translocated material provides the same triple gene dosage as in trisomy 21. Such cases are frequently, but not always, familial, and the translocated chromosome is inherited from one of the parents, usually the mother, who is a carrier of a Robertsonian translocation, for example. A mother with karyotype 45XX, DIR1421. In cells with Robertsonian translocations, the genetic material, normally found on long arms of 2 pairs of chromosomes, is distributed among only 3 chromosomes. This affects chromosome pairing during meiosis, and as a result, the gametes have a high probability of being aneuploid. Approximately 1% of patients with Down syndrome are mosaics, having a mixture of cells with 46 or 47 chromosomes. This mosaicism results from mitotic non-disjunction of chromosome 21 during an early stage of embryogenesis. Symptoms in such cases are variable and milder, depending on the proportion of abnormal cells. Clearly, in cases of translocation or mosaic Down syndrome, maternal age is of no importance. The diagnostic clinical features of this condition, flat facial profile, oblique palpebral fissures, and epicanthic folds, figure 5.20, are usually readily evident even at birth. Down syndrome is a leading cause of severe intellectual disability. It should be pointed out that some mosaics with Down syndrome have mild phenotypic changes and may even have normal or near normal intelligence. In addition to the phenotypic abnormalities and the intellectual disability already noted, some other clinical features are worthy of note. Approximately 40% of the patients have congenital heart disease. The most frequent forms of congenital heart diseases in Down syndrome are atrioventricular septal defects, constituting 43% of cases, whereas ventricular septal defects, atrial septal defects, and tetralogy of Fallot constitute 32%, 19%, and 6% respectively. Cardiac problems are responsible for the majority of the deaths in infancy and early childhood. Several other congenital malformations, including atresias of the esophagus and small bowel, are also common. Children with trisomy 21 have a high risk of developing leukemia. There is 20-fold increased risk of developing acute B lymphoblastic leukemias and 500-fold increased risk of acute myeloid leukemias. The latter, most commonly, is acute megakaryoblast leukemia. Virtually all patients with trisomy 21, older than age 40, develop neuropathologic changes characteristic of Alzheimer's disease, a degenerative disorder of the brain. Patients with Down syndrome have abnormal immune responses that predispose them to serious infections, particularly of the lungs, and to thyroid autoimmunity. Although several abnormalities, affecting mainly T-cell functions, have been reported, the basis of immunologic disturbances is not clear. Despite all these problems, improved medical care has increased the longevity of individuals with trisomy 21. Currently, the median age at death is 47 years, up from 25 years in 1983. Although the long arm of chromosome 21 was fully sequenced in 2000s, progress in unraveling the molecular basis of Down syndrome has been quite slow. This stems in part from the fact that Down syndrome results from gene dosage imbalance rather than the action of a few genes. A sampling of some of the observations follows. Gene dosage. The majority of the protein coding genes mapped to chromosome 21 are overexpressed. Included among these is the gene for amyloid beta precursor protein AP. As discussed in Chapter 28, aggregation of amyloid beta proteins is the critical initiating event in the development of Alzheimer's disease and could contribute to the early onset of Alzheimer's disease that occurs in individuals with Down syndrome. Mitochondrial dysfunction. Approximately 10% of the genes overexpressed in Down syndrome are directly or indirectly involved in regulation of mitochondrial functions. In keeping with this, mitochondria are abnormal both morphologically and functionally in several tissues. For example, cristae are broken or swollen. There is evidence of mitochondrial stress with generation of reactive oxygen species and activation of apoptosis. Non-coding RNAs. Chromosome 21 has the highest density of L and C RNAs whose target sequences are widely expressed. 168 Chapter 5 Genetic Disorders. Intellectual disability. Abundant neck skin. Congenital heart defects. Intestinal stenosis. Epicanthic folds and flat facial profile. Simian crease, umbilical hernia predisposition to leukemia, hypotonia. Trisomy 21, Down syndrome incidence, 1 in 700 births. Karyotypes. Trisomy 21 type, 47XX, 21 translocation type, 46E1 translocation type, trisomy 21 translocation type. Prominent occiput, intellectual disability, micrognathia, gap between first and second toe low set ears, short neck, overlapping fingers. Trisomy 18, Edwards syndrome. Incidence, 1 in 8,000 births. Karyotypes. Trisomy 18 type, 47XX, 18 mosaic type, 46X47XX, 18. Congenital heart defects, renal malformations, limited hip abduction, microphthalmia polydactyly, microcephaly and intellectual disability, cleft lip and palate rocker bottom feet, cardiac defects, umbilical hernia renal defects, trisomy 13 Patau syndrome. Incidence, 1 in 15,000 births. Karyotypes, trisomy 13 type, 47XX, 13 translocation type, 46X13 DIR 1347, 13 type, 4 rocker bottom feet, figure 5. Clinical features and karyotypes of selected autosomal trisomies. Disorders 169. On other chromosomes, making the task of identification of genes whose products dictate the phenotype of trisomy 21 exceedingly difficult. Much progress is being made in the molecular diagnosis of Down syndrome prenatally. Approximately 5% to 10% of the total cell-free DNA in maternal blood is derived from the fetus and can be identified by polymorphic genetic markers. By using next-generation sequencing, the gene dosage of chromosome 21 linked genes in fetal DNA can be determined with great precision. This is a powerful non-invasive screening test for prenatal diagnosis of trisomy 21, as well as other trisomies. Most laboratories require confirmation of a positive screening test with conventional karyotyping. Other trisomies. A variety of other trisomies involving chromosomes 8, 9, 13, 18, and 22 have been described. Only trisomy 18 Edwards syndrome and trisomy 13 Patau syndrome are common enough to merit brief mention here. As noted in figure 5.20, they share several karyotypic and clinical features with trisomy 21. Thus, most cases result from meiotic non-disjunction and therefore carry a complete extra copy of chromosome 13 or 18. As in Down syndrome, an association with increased maternal age is also noted. In contrast to trisomy 21, however, the malformations are much more severe and wide-ranging. As a result, only rarely do infants survive beyond the first year of life. Most succumb within a few weeks to months. Chromosome 22q11.2, deletion syndrome. Chromosome 22q11.2. Deletion syndrome encompasses a spectrum of disorders that result from a small deletion of band Q11.2 on the long arm of chromosome 22. The syndrome is fairly common, occurring in as many as 1 in 4,000 births, but it is often missed because of variable clinical features. These include congenital heart defects, abnormalities of the palate, facial dysmorphism, developmental delay, and variable degrees of T-cell immunodeficiency and hypocalcemia. Previously, these clinical features were considered to represent two different disorders. DiGeorge syndrome and velocardiofacial syndrome. In a very small number of cases, there is a deletion of 10p13 to 14. Patients with DiGeorge syndrome have thymic hypoplasia with resultant T-cell immunodeficiency chapter 6, parathyroid hypoplasia giving rise to hypocalcemia, a variety of cardiac malformations affecting the outflow tract, and mild facial anomalies. Atopic disorders, e.g., allergic rhinitis, and autoimmunity, e.g., thrombocytopenia, may also be seen. The clinical features of the so-called velocardiofacial syndrome include facial dysmorphism, prominent nose, retrognathia, cleft palate, cardiovascular anomalies, and learning disabilities. Less frequently, these patients also have immunodeficiency. Despite the overlapping clinical features of these two conditions, e.g., cardiac malformations, facial dysmorphology, it was only after these two apparently unrelated syndromes were found to be associated with a similar cytogenetic abnormality that the clinical overlap came into focus. Recent studies indicate that, in addition to the numerous structural malformations, individuals with the 22q11.2 deletion syndrome are at a particularly high risk for psychotic illnesses, such as schizophrenia and bipolar disorders. In fact, it is estimated that schizophrenia develops in approximately 25% of adults with this syndrome. Conversely, deletions of the region can be found in 2% to 3% of individuals with childhood-onset schizophrenia. In addition, attention deficit hyperactivity disorder is seen in 30% to 35% of affected children. The diagnosis of this condition may be suspected on clinical grounds, but can be established only by detection of the deletion by FISH figure 521. By this test, approximately 90% of individuals previously diagnosed as having DiGeorge syndrome and 80% of those with a velocardiofacial syndrome have a deletion of 22q11.2. 30% of individuals with conotruncal cardiac defects but no other features of this syndrome also reveal deletions of the same chromosomal region. The molecular basis of this syndrome is not fully understood. The deleted region is large, approximately 1.5 MB, and includes 30 to 40 genes. The clinical heterogeneity, with predominant immunodeficiency, in some cases DiGeorge syndrome, and predominant dysmorphology, and cardiac malformations in other cases, probably reflects the variable position and size of the deleted segment from this genetic region. Approximately 30 candidate genes have been mapped to the deleted region. Among these, TBX1, a T-box transcription factor, is most closely associated with the phenotypic features of this syndrome. This gene is expressed in the pharyngeal mesenchyme and endodermal pouch, from which facial structures, thymus, and parathyroid are derived. The targets of TBX1 include PAX9, a gene that controls the development of the palate, parathyroids, and thymus. Clearly, there are other genes that contribute to the behavioral and psychiatric disorders that remain to be identified. Figure 5.21. Fluorescence in situ hybridization of both metaphase chromosomes and an interphase cell from a patient with DiGeorge syndrome demonstrating the deletion of a probe that maps to chromosome 22q11.2. The 22q11.2 probe is in red and the control probe, localized to 22q, is in green. The metaphase spread shows one chromosome 22 with both a green signal control probe and a red signal from the 22q11.2 probe. The other chromosome 22 shows only hybridization with the control probe green, but no red 22q11.2 signal since there is a deletion on this chromosome. The interphase cell also shows a hybridization pattern consistent with a deletion of chromosome 22q11.2. Courtesy Dr. Stuart Schwartz, Department of Pathology, University of Chicago, Chicago, Ill. Chromosomal. Chapter 5. Genetic disorders. Key concepts: cytogenetic disorders involving autosomes. Down syndrome is associated with an extra copy of genes on chromosome 21, most commonly due to trisomy 21, and less frequently from translocation of extra chromosomal material from chromosome 21 to other chromosomes or from mosaicism. Patients with Down syndrome have severe intellectual disability, flat facial profile, epicanthic folds, cardiac malformations, higher risk of leukemia and infections, and premature development of Alzheimer's disease. Deletion of genes at chromosomal locus 22q11.2 gives rise to malformations affecting the face, heart, thymus, and parathyroids. The resulting disorders are recognized as DiGeorge syndrome, thymic hypoplasia with diminished T-cell immunity, and parathyroid hypoplasia with hypocalcemia, and velocardiofacial syndrome, congenital heart disease involving outflow tracts, facial dysmorphism, and developmental delay. 170. Cytogenetic disorders involving sex chromosomes. Genetic diseases associated with changes involving the sex chromosomes are far more common than those related to autosomal aberrations. Furthermore, imbalances, excess, or loss of sex chromosomes are much better tolerated than are similar imbalances of autosomes. In large part, this latitude relates to two factors that are peculiar to the sex chromosomes. 1, lionization or inactivation of all but one X chromosome, and 2, the modest amount of genetic material carried by the Y chromosome. These features are discussed briefly in relation to sex chromosomal disorders. In 1961, Mary Lyon outlined the idea of X inactivation, now commonly known as the Lyon hypothesis. It states that 1, only one of the X chromosomes is genetically active. 2, the other X chromosome of either maternal or paternal origin undergoes heteropyknosis and is rendered inactive. Inactivation of either the maternal or the paternal X chromosome occurs at random among all the cells of the blastocyst on or about day 5.5 of embryonic life. And 4, inactivation of the same X chromosome persists in all the cells derived from each precursor cell. Thus, the great preponderance of normal females are in reality mosaics and have two populations of cells, one with an inactivated maternal X chromosome and the other with an inactivated paternal X chromosome. Herein lies the explanation of why females have the same dosage of X-linked active genes as males. The inactive X chromosome can be seen in the interphase nucleus as a darkly staining small mass in contact with the nuclear membrane known as the Barr body or X chromatin. The molecular basis of X inactivation involves a unique gene called ESIS, whose product is a lncRNA chapter 1 that is retained in the nucleus, where it coats the X chromosome that it is transcribed from and initiates a gene silencing process by chromatin modification and DNA methylation. The XIST allele is switched off in the active X chromosome. Although it was initially thought that all the genes on the inactive X chromosome are switched off, it is now established that many genes escape X inactivation. Molecular studies suggest that 30% of genes on XP and a smaller number, 3%, on XQ escape X inactivation. At least some of the genes that are expressed from both X chromosomes are important for normal growth and development. This notion is supported by the fact that patients with monosomy of the X chromosome, Turner syndrome 45X, have severe somatic and gonadal abnormalities. If a single dose of X-linked genes were sufficient, no detrimental effect would be expected in such cases. Furthermore, although one X chromosome is inactivated in all cells during embryogenesis, it is selectively reactivated in oogonia before the first meiotic division. Thus, it seems that both X chromosomes are required for normal growth as well as oogenesis. The tips of short and long arms of X and Y chromosomes have regions of homology that recombine during meiosis and are therefore inherited as autosomal loci. For this reason, they are called pseudo-autosomal regions. These genes also escape X inactivation. These mechanisms ensure that males and females have equivalent doses of genes that map on X and Y chromosomes. With respect to the Y chromosome, it is well known that this chromosome is both necessary and sufficient for male development. Regardless of the number of X chromosomes, the presence of a single Y chromosome determines the male sex. The gene that dictates testicular development, SRI, sex-determining region Y gene, is located on its distal short arm. For quite some time, this was considered to be the only gene of significance on the Y chromosome. Recent studies, however, have yielded a rich harvest of several gene families in the so-called male-specific Y region, or MSY region, harboring 75 protein-coding genes. All of these are believed to be testis-specific and are involved in spermatogenesis. In keeping with this, all Y chromosome deletions are associated with azoospermia. By comparison, the X chromosome has 840 coding genes. The following features are common to all sex chromosome disorders. In general, sex chromosome disorders cause subtle, chronic problems relating to sexual development and fertility. Sex chromosome disorders are often difficult to diagnose at birth, and many are first recognized at the time of puberty. In general, the greater the number of X chromosomes in both males and females, the greater the likelihood of intellectual disability. The two most important disorders arising in aberrations of sex chromosomes are described briefly here. Klinefelter syndrome. Klinefelter syndrome is best defined as male hypogonadism that occurs when there are two or more X chromosomes and one or more Y chromosomes. It is one of the most frequent forms of genetic diseases involving the sex chromosomes, as well as one of the most common causes of hypogonadism in males. The incidence of this condition is reported to be approximately 1 in 660 live male births. This is an underestimate since Klinefelter syndrome has a range of phenotypic manifestations. and those with mild features are never seen by healthcare providers. The clinical features of Klinefelter syndrome can be attributed to two major factors: one, aneuploidy, and the impact of increased gene. Chromosomal disorders 171. Dosage by the supernumerary X, and 2, the presence of hypogonadism. Klinefelter syndrome is rarely diagnosed before puberty, particularly because manifestations of hypogonadism do not develop before early puberty. Most patients have a distinctive body habitus with an increase in length between the soles and the pubic bone, which creates the appearance of an elongated body. Also characteristic are unicoid body habitus with abnormally long legs, small atrophic testes often associated with a small penis, and lack of such secondary male characteristics as deep voice, beard, and male distribution of pubic hair. Gynecomastia may be present. The cognitive abilities range from average to below average, with modest deficit in verbal skills, particularly those that are used in reading and language comprehension. Patients with Klinefelter syndrome develop several comorbid conditions. There is increased incidence of type 2 diabetes and the metabolic syndrome that gives rise to insulin resistance. Patients are at a higher risk for congenital heart disease, particularly mitral valve prolapse, which is seen in about 50% of adults. In addition, there is a higher prevalence of atrial and ventricular septal defects. There is also an increased incidence of osteoporosis and fractures due to sex hormonal imbalance. Patients with Klinefelter syndrome have a 20 to 30-fold higher risk of developing extragonadal germ cell tumors, mostly mediastinal teratomas. In addition, breast cancer and autoimmune diseases such as systemic lupus erythematosus occur more frequently. It should be noted that the physical attributes described here are quite variable, the only consistent finding being hypogonadism. Klinefelter syndrome is an important genetic cause of reduced spermatogenesis and male infertility. In some patients, the testicular tubules are totally atrophied and replaced by pink, hyaline collagenous ghosts. In others, apparently normal tubules are interspersed with atrophic tubules. In some patients, all tubules are primitive and appear embryonic, consisting of cords of cells that never developed a lumen or progressed to mature spermatogenesis. Leydig cells appear prominent as a result of the atrophy and crowding of the tubules and elevation of gonadotropin concentrations. Plasma gonadotropin concentrations, particularly follicle-stimulating hormone FSH, are consistently elevated, whereas testosterone levels are variably reduced. Mean plasma estradiol levels are elevated by an as-yet unknown mechanism. The ratio of estrogens and testosterone determines the degree of feminization in individual cases. Classic Klinefelter syndrome is associated with a 47XXY karyotype 90% of cases. This complement of chromosomes results from non-disjunction during the meiotic divisions in the germ cells of one of the parents. Maternal and paternal non-disjunction at the first meiotic division is roughly equally involved. There is no phenotypic difference between those who receive the extra X chromosome from their father and those who receive it from their mother. Advanced maternal age, greater than 40 years, is a risk factor. In addition to this classic karyotype, approximately 15% of patients with Klinefelter syndrome have a variety of mosaic patterns, most of them being 46XY47. That's XY. In some, there is a cell line with structurally abnormal X chromosome, e.g., 47IXQY. As is the case with normal females, all but one X chromosome undergoes inactivation in patients with Klinefelter syndrome. Why then do the patients with this disorder have hypogonadism? And associated features? The explanation for this lies in genes on the X chromosome that escape lionization and in the pattern of X inactivation. One pathogenic mechanism is related to uneven dosage compensation during X inactivation. It is now known that about 35% of the X-linked genes escape inactivation. Thus, there is an extra dose of these genes compared with normal males in whom only one copy of the X chromosome is active. It appears that overexpression of one or more of these genes leads to hypogonadism. A similar mechanism may also dictate some somatic features. The short stature homeobox SHOX gene that maps on the pseudo-autosomal region of XP is one of the genes that is not subject to X inactivation. An extra copy of this growth-related gene is probably responsible for the tall stature and long legs typical of Klinefelter syndrome. It should be noted that most genes, whose expression is upregulated in Klinefelter syndrome, lie outside the X chromosome. This implies that the supernumerary X chromosome can regulate gene expression on autosomes. A second mechanism involves the gene encoding the androgen receptor, through which testosterone mediates its effects. The androgen receptor gene maps to the X chromosome and contains highly polymorphic CAG trinucleotide, repeats. The functional response of the receptor to any particular dose of androgen is dictated in part by the number of KAG repeats, as receptors with shorter KAG repeats are more sensitive to androgens than those with long KAG repeats. In persons with Klinefelter syndrome, the X chromosome bearing the androgen receptor allele with the shortest KAG repeat is preferentially inactivated. In XXY males with low testosterone levels, expression of androgen receptors with long KAG repeats exacerbates the hypogonadism and appears to account for certain aspects of the phenotype, such as small penis size. Turner syndrome. Turner syndrome results from complete or partial monosomy of the X chromosome and is characterized by hypogonadism in phenotypic females. It is the most common sex chromosome abnormality in females, affecting about 1 in 2,000 live-born females. With routine cytogenetic methods, 3 types of karyotypic abnormalities are seen in individuals with Turner syndrome. Approximately 57% are missing an entire X chromosome, resulting in a 45X karyotype. Of the remaining 43%, approximately 1/3, 14%, have structural abnormalities of the X chromosomes, and 2/3, 29%, are mosaics. The common feature of the structural abnormalities is to produce partial monosomy of the X chromosome. In order of frequency, the structural abnormalities of the X chromosome include 1, an isochromosome of the long arm, 46XXQ10, resulting in the loss of the short arm, 2, deletion of portions of both long and short arms, resulting in the formation of a ring chromosome, 46XRX, and 3, deletion of portions of the short or long arm, 46 times del XQ, or 46 times del XP. The mosaic patients have a 45X cell population along with 1 or more karyotypically normal or abnormal cell types. 172. Chapter 5. Genetic disorders. Examples of karyotypes that mosaic Turner females may have are the following: 1, 45X, 46XX, 245, 46XY, or 45 are the following. Studies suggest that the prevalence of mosaicism in Turner syndrome may be much higher than the 30% detected by conventional cytogenetic studies. With the use of more sensitive techniques, the prevalence of mosaic Turner syndrome increases to 75%. Because 99% of conceptuses with an apparent 45X karyotype are non-viable, many authorities believe that there are no truly non-mosaic Turner syndrome patients. While this issue remains controversial, it is important to appreciate the karyotypic heterogeneity associated with Turner syndrome because it is responsible for significant variations in phenotype. In patients in whom the proportion of 45X cells is high, the phenotypic changes are more severe than in those who have readily detectable mosaicism. The latter may have an almost normal appearance and may present only with primary amenorrhea. A very small number of patients are able to conceive. 5% to 10% of patients with Turner syndrome have Y chromosome sequences either as a complete Y chromosome, e.g. 45X46, XY karyotype, or as fragments of Y chromosomes translocated on other chromosomes. These patients are at a higher risk for development of a gonadal tumor, gonadoblastoma. The most severely affected patients generally present during infancy with edema of the dorsum of the hand and foot due to lymph stasis, and sometimes swelling of the nape of the neck. The latter is related to markedly distended lymphatic channels, producing a so-called cystic hygroma, Chapter 10. As these infants develop, the swellings subside but often leave bilateral neck webbing and persistent looseness of skin on the back of the neck. Congenital heart disease is also common, affecting 25% to 50% of patients. Left-sided cardiovascular abnormalities, particularly productal coarctation of the aorta and bicuspid aortic valve, are seen most frequently. Approximately 5% of young women initially diagnosed with coarctation of aorta have Turner syndrome. Aortic root dilatation is present in 30% of cases, and there is a 100-fold higher risk of aortic dissection. Cardiovascular abnormalities are the most important cause of increased mortality in children with Turner syndrome. The principal clinical features in adolescents and adults are illustrated in Fig. 5.22. At puberty, there is failure to develop normal secondary sex characteristics. The genitalia remain infantile, breast development is inadequate, and there is little pubic hair. The mental status of patients is usually normal, but subtle defects in nonverbal, visual spatial information processing have been noted. Of particular importance in establishing the diagnosis in an adult is the shortness of stature, rarely exceeding 150 centimeters in height, and amenorrhea. Turner syndrome is the single most important cause of primary, low posterior hairline webbing of neck, broad chest, and widely spaced nipples, short stature, coarctation of aorta, cubitus valgus, streak ovaries, infertility, amenorrhea, Turner syndrome incidence, 1 in 3,000 female births, karyotypes, classic, 45X defective, second X chromosome, 46XIXQ, 46X del XQ, 46X del XP, 46XRX, mosaic type, 45X46XX, 45X46XY, 45X47XX, 45X46X, Q10. 4 at birth. Figure 5.2. Two clinical features and karyotypes of Turner syndrome. Single gene disorders with non-classic inheritance, 173. Amenorrhea, accounting for approximately 1/3 of the cases. For reasons that are not clear, approximately 50% of patients develop autoantibodies that react with the thyroid gland, and up to half of these develop clinically manifest hypothyroidism. Equally mysterious is the presence of glucose intolerance, obesity, non-alcoholic fatty liver disease, and insulin resistance in a subset of patients. Some have full-fledged metabolic syndrome. The occurrence of insulin resistance is significant because therapy with growth hormone, commonly used in these patients, worsens insulin resistance. The molecular pathogenesis of Turner syndrome is not completely understood, but studies have begun to shed some light. In approximately 80% of cases, the X chromosome is maternal in origin, suggesting that there is an abnormality in paternal gametogenesis. As mentioned earlier, both X chromosomes are active during oogenesis and are essential for normal development of the ovaries. During normal fetal development, ovaries contain as many as 7 million oocytes. The oocytes gradually disappear so that by menarche, their numbers have dwindled to a mere 400,000, and when menopause occurs, fewer than 10,000 remain. In Turner syndrome, fetal ovaries develop normally early during the first 18 weeks of gestation, but the absence of the second X chromosome leads to an accelerated loss of oocytes, which is complete by age 2 years. In a sense, therefore, menopause occurs before menarche, and the ovaries are reduced to atrophic fibrous strands, devoid of ova and follicles, streak ovaries. Studies of patients with Turner syndrome with deletions affecting the short or long arm have revealed that many of the somatic features are determined by genes on the short arm, whereas genes on the long arm affect fertility and menstruation. Among the genes involved in the Turner phenotype is the SHOX gene at XP22.33. As mentioned earlier, SHOX maps at the pseudo-autosomal region of the X and Y chromosomes and escapes X inactivation. Thus, both normal males and normal females have 2 copies of this gene. Haploinsufficiency of SHOX in Turner syndrome is believed to give rise to short stature. Indeed, deletions of the SHOX gene are noted in 2% to 5% of otherwise normal children with short stature. In keeping with its role as a critical regulator of growth, SHOX is expressed during fetal life in the growth plates of several long bones, including the radius, ulna, tibia, and fibula. It is also expressed in the first and second pharyngeal arches. Just as the loss of SHOX is always associated with short stature, excess copies of this gene in Klinefelter syndrome are associated with tall stature. Whereas haploinsufficiency of SHOX can explain growth deficit in Turner syndrome, it cannot explain other clinical features such as cardiac malformations and metabolic abnormalities. Clearly, several other genes located on the short arm of X chromosome are involved. Growth hormone and estradiol are used to treat Turner syndrome with a reasonable degree of success. Hermaphroditism and pseudohermaphroditism. The problem of sexual ambiguity is exceedingly complex, and only limited observations are possible here. For more details, the reader should refer to specialized sources. It will be no surprise to medical students that the sex of an individual can be defined on several levels. Genetic sex is determined by the presence or absence of a Y chromosome. No matter how many X chromosomes are present, a single Y chromosome dictates testicular development and the genetic male sex. The initially indifferent gonads of both male and female embryos have an inherent tendency to feminize, unless influenced by Y chromosome-dependent masculinizing factors. Gonadal sex is based on the histologic characteristics of the gonads. Ductal sex depends on the presence of derivatives of the Müllerian or Wolffian ducts. Phenotypic, or genital, sex is based on the appearance of the external genitalia. Sexual ambiguity is present whenever there is disagreement among these various criteria for determining sex. The term true hermaphrodite implies the presence of both ovarian and testicular tissue. In contrast, a pseudohermaphrodite represents a disagreement between the phenotypic and gonadal sex, i.e., A female pseudohermaphrodite has ovaries, but male external genitalia. A male pseudohermaphrodite has testicular tissue, but female-type genitalia. The genetic bases of these conditions are quite variable and beyond the scope of our discussion here. Key concepts: cytogenetic disorders involving sex chromosomes. In females, one X chromosome, maternal or paternal, is randomly inactivated during development, Lyon hypothesis. In Klinefelter syndrome, there are 2 or more X chromosomes, with one Y chromosome as a result of non-disjunction of sex chromosomes. Patients have testicular atrophy, sterility, reduced body hair, gynecomastia, and unicoid body habitus. It is the most common cause of male sterility. In Turner syndrome, there is partial or complete monosomy of genes on the short arm of the X chromosome, most commonly due to absence of one X chromosome 45X mosaicism or deletions involving the short arm of the X chromosome. Short stature, webbing of the neck, cubitus valgus, cardiovascular malformations, amenorrhea, lack of secondary sex characteristics, and fibrotic ovaries are typical clinical features. Single gene disorders with non-classic inheritance. It has become increasingly evident that transmission of certain single gene disorders does not follow classic Mendelian principles. This group of disorders can be classified into 4 categories: diseases caused by trinucleotide repeat mutations, disorders caused by mutations in mitochondrial genes, disorders associated with genomic imprinting, disorders associated with gonadal mosaicism. Clinical and molecular features of some single gene diseases that exemplify non-classic patterns of inheritance are described next. Diseases caused by trinucleotide repeat mutations. Expansion of trinucleotide repeats is an important genetic cause of human disease, particularly neurodegenerative. 174th Chapter 5 Genetic Disorders. 8.5 Telba of trinucleotide repeat disorders. Locus. Disease gene. Expansions affecting non-coding regions. Fragile X syndrome. fMRI. Fraxa. Friedreich ataxia. FXN. Myotonic dystrophy DMPK. XQ27.3. 9Q21.1. 19Q13.3. Expansions affecting coding regions. Spinobulbar muscular AR atrophy Kennedy disease. Huntington disease HTT. Dentatorubral pallidoluysian ATNL atrophy Hall River syndrome. Spinocerebellar ataxia type 1 ATXN1. Spinocerebellar ataxia type 2 ATXN2. Spinocerebellar ataxia type 3 ATXN3. Machado-Joseph disease. Spinocerebellar ataxia type 6 ataxin 6. XQ12, 4P16.3, 12P13.31, 6P2312Q24.1, 14Q21, 19P13.3, spinocerebellar ataxia, type 7 3P14.1 ataxin to 7. Protein, FMR1 protein FMRP frataxin myotonic dystrophy, protein kinase DMPK, androgen receptor, AR, Huntington, atrophin 1. Ataxin 1, Ataxin 2, Ataxin 3. ALPH, A1, A voltage-dependent calcium channel subunit. Ataxin 7, repeat, CGG, GAACTG. CAG-G, CAG, 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 CAG. CAG, CAG, normal. 6, 55, 7, 34, 537, 534, 635, 7, 35, 644, 13, 33, 12, 40, 4, 18, 4, 35, no. Repeats disease, 55, 200, greater, full 34. Greater than 237, 70. 39, 250. 49, 88. Greater than 39, greater than 31, 55, 84. 21, 33. 37, 306 examples. Disorders. The discovery in 1991 of expanding trinucleotide repeats as a cause of Fragile X syndrome, FXS, was a landmark in human genetics. Since then, the origins of about 40 human diseases, Table 5.8, have been traced to unstable nucleotide repeats, and the number continues to grow. All the disorders discovered so far are associated with neurodegenerative changes. Some general principles apply to these diseases. The causative mutations are associated with the expansion of a stretch of trinucleotides that usually share the nucleotides G and C. In all cases, the DNA is unstable, and an expansion of the repeats above a certain threshold impairs gene function in various ways, discussed later. In recent years, diseases associated with unstable tetranucleotides, pentanucleotides, and hexanucleotides have also been found, establishing this as a fundamental mechanism of neuromuscular diseases. The proclivity to expand depends strongly on the sex of the transmitting parent. In FXS, expansions occur during oogenesis, whereas in Huntington disease they occur during spermatogenesis. There are three key mechanisms by which unstable repeats cause diseases. 1. Loss of function of the affected gene, typically by transcription silencing. As in FXS, in such cases the repeats are generally in the non-coding part of the gene. A toxic gain of function by alterations of protein structure. As in Huntington disease and spinocerebellar ataxias, in such cases the expansions occur in the coding regions of the genes. A toxic gain of function mediated by RNA, as is seen in Fragile X-associated tremor, ataxia syndrome. As in FXS, the non-coding parts of the gene are affected, figure 5.23. The pathogenetic mechanisms underlying disorders caused by mutations that affect coding regions seem to be distinct from those in which the expansions affect non-coding regions. The former usually involve KAG repeats coding for polyglutamine tracts in the corresponding proteins. Such polyglutamine diseases are characterized by progressive neurodegeneration, typically striking in midlife. Polyglutamine expansions lead to toxic gain of function, whereby the abnormal protein may interfere with the function of the normal protein, a dominant negative activity, or acquire a novel pathophysiologic toxic activity. The precise mechanisms by which expanded polyglutamine proteins cause disease are not fully understood. In most cases, the proteins are misfolded and tend to aggregate. The aggregates may suppress transcription of other genes, cause mitochondrial dysfunction, or trigger the unfolded protein stress response and apoptosis, chapters 1 and 2. A morphologic hallmark of these diseases is the accumulation of aggregated mutant proteins in large intranuclear inclusions. While formation of aggregates is common to many polyglutamine diseases, evidence of a direct toxic role of aggregates is not universal. In fact, some observers believe that aggregation may be protective by sequestration of the misfolded protein. Other models of pathogenicity implicate downstream effects mediated by proteolytic fragments of the polyglutamine fragment. Much more needs to be learned before therapeutic strategies can be developed. Fragile X syndrome, FXS. FXS is the most common genetic cause of intellectual disability in males, and overall the second most common cause after Down syndrome. It results from a trinucleotide expansion mutation in the familial mental retardation 1 FMR1 gene. Although discovered initially as the cause of FXS, expansion mutations affecting the FMR1 gene are now known to be present in two other well-defined disorders: Fragile X-associated tremor, ataxia syndrome, and Fragile X-associated primary ovarian insufficiency. We begin our discussion of these disorders with consideration of FXS. Disorders with non-classic inheritance 175. 5 feet. Promoter, UTR, intron, exon, UTR. 3 feet. Expansions. Sequences, mechanisms, disease, transcriptional, silencing, loss of protein, function, Fragile X syndrome. CGG triplet. Transcriptional dysregulation. Accumulation of toxic mRNA. Fragile X tremor ataxia. GAA triplet transcriptional, silencing, loss of protein, function, Friedreich ataxia. CAG triplet polyglutamine expansions with misfolding. Toxic gain of function, Huntington disease, Fragile X associated, primary ovarian insufficiency, figure 5.23 sites of expansion, and the affected sequence in selected diseases caused by nucleotide repeat mutations. UTR, untranslated region. FXS has a frequency of 1 in 1,550 for affected males and 1 in 8,000 for affected females. Its name derives from an inducible cytogenetic abnormality in the X chromosome within which the FMR1 gene maps. The cytogenetic alteration was discovered as a discontinuity of staining or as a constriction in the long arm of the X chromosome when cells are cultured in a folate-deficient medium. Because it appears that the chromosome is broken at this locale, it was named as a fragile site, figure 524. This method of detection has now been supplanted by DNA-based analysis of triplet repeat size, as discussed later. Males with FXs have marked intellectual disability. They have a characteristic physical phenotype that includes a long face with a large mandible, large everted ears, and large testicles, macroorchidism, hyperextensible joints, a high arched palate, and mitral valve prolapse noted in some patients mimic a connective tissue disorder. These and other physical abnormalities described in this condition, however, are not always present and, in some cases, are quite subtle. The most distinctive feature is macroorchidism, which is observed in at least 90% of affected postpubertal males. In addition to intellectual disability, several neurologic and neuropsychiatric manifestations have been recognized in patients with FXS. These include epilepsy in 30% of cases, aggressive behavior in 90% of cases. Autism spectrum disorder includes several conditions such as autism and Figure 524 Fragile X, seen as discontinuity of staining. Courtesy Dr. Patricia Howard Peebles, University of Texas Southwestern Medical Center, Dallas, Texas. Single gene, Asperger syndrome, and anxiety disorder, hyperactivity disorder. The latter two affect 50% to 75% of males with FXS. 2% to 5% of patients diagnosed first with non-syndromic autism have a mutation in the FMR1 gene. As with other X-linked diseases, FXS affects males predominantly. Analysis of several pedigrees, however, reveals some patterns of transmission not typically associated with other X-linked recessive disorders. Figure 5.25. Carrier males, approximately 20% of males who, by pedigree analysis and by molecular tests, are known to carry a Fragile X mutation, are clinically normal. Because carrier males transmit the trait through all their phenotypically normal daughters to affected grandchildren, they are called normal transmitting males. Affected females, 30% to 50% of carrier females are affected, i.e., have intellectual disability as well as other features described here, a number much higher than that in other X-linked recessive disorders. Risk of phenotypic effects. Risk depends on the position of the individual in the pedigree. For example, brothers of transmitting males are at a 9% risk of having intellectual disability, whereas grandsons of transmitting males incur a 40% risk. Anticipation. This refers to the observation that clinical features of FXS worsen with each successive generation, as if the mutation becomes increasingly deleterious as it is transmitted from a man to his grandsons and great-grandsons through daughters. The first breakthrough in resolving these perplexing observations came when linkage studies localized the mutation responsible for this disease to XQ27.3 within the cytogenetically abnormal region. At this locale lies the FMR1 gene, characterized by multiple tandem repeats of the nucleotide sequence CGG in its 5 untranslated region. In the normal population, the number of CGG repeats is small, ranging from 6 to 55, average 29. The presence and severity of clinical symptoms is related to the amplification of the CGG repeats. Thus, normal transmitting males and carrier females carry 55 to 200 CGG repeats. Expansions of this size are called permutations. In contrast, patients with FXS. Chapter 5. Genetic disorders. Carrier male, normal female. X chromosomes phenotype. Permutation. Normal. 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 Carrier female unrelated. X chromosomes phenotype. Normal. Normal. Normal permutation. Normal. 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 Normal permutation. Normal. 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 X chromosomes phenotype. Full mutation. Affected. Normal. Full mutation. 50% of females affected, often mildly. Normal. 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 Figure 5.25 Fragile X pedigree. Note that in the first generation, all sons are normal and all females are carriers. During oogenesis in the carrier female, permutation expands to full mutation. Hence, in the next generation, all males who inherit the X with full mutation are affected. However, only 50% of females who inherit the full mutation are affected, and only mildly. Not shown are Fragile X-associated ataxia, tremor, and Fragile X-associated primary ovarian failure that can occur in permutation careers. Courtesy Dr. Nancy Schneider, Department of Pathology, University of Texas Southwestern Medical Center, Dallas, Texas. 176th. Have an extremely large expansion of the repeat region, 200 to 4,000 repeats, or full mutations. Full mutations are believed to arise by further amplification of the CGG repeats seen in permutations. How this process takes place is quite peculiar. Carrier males transmit the repeats to their progeny with small changes in repeat number. When the permutation is passed on by a carrier female, however, there is a high probability of a dramatic amplification of the CGG repeats, leading to intellectual disability in most male offspring and 50% of female offspring. Thus, it seems that during the process of oogenesis, but not spermatogenesis, permutations can be converted to mutations by triplet repeat amplification. This explains the unusual inheritance pattern. That is, the likelihood of intellectual disability is much higher in grandsons than in brothers of transmitting males because grandsons incur the risk of inheriting a permutation. from their grandfather that is amplified to a full mutation in their mother's ova. By comparison, brothers of transmitting males, being higher up in the pedigree, are less likely to have a full mutation. These molecular details also provide a satisfactory explanation of anticipation, a phenomenon that remained unexplained until triplet repeat mutations were identified. Why only 50% of the females with the full mutation are clinically affected is not clear. Presumably in those who are clinically affected, there is unfavorable lionization, i.e., there is a higher frequency of cells in which the X chromosome carrying the mutation is active. The molecular basis of intellectual disability and other somatic changes is related to loss of function of the fragile X mental retardation protein, FMRP, the product of FMR1 gene. As mentioned earlier, the normal FMR1 gene contains up to 55 CGG repeats in its 5 untranslated region. When the trinucleotide repeats in the FMR1 gene exceed approximately 230, the DNA of the entire 5 region of the gene becomes abnormally methylated. Methylation also extends upstream into the promoter region of the gene, resulting in transcriptional suppression of FMR1. The resulting absence of FMRP is believed to cause the phenotypic changes. FMRP is a widely expressed cytoplasmic protein, most abundant in the brain and testes, the 2 organs most affected in this disease. Its proposed functions in the brain are the following. FMRP selectively binds mRNA as associated with polysomes and regulates their intracellular transport to dendrites. Unlike other cells in neurons, protein synthesis occurs both in the perinuclear cytoplasm and in dendritic spines. Newly made FMRP translocates to the nucleus, where it assembles into a complex containing mRNA transcripts that encode presynaptic and postsynaptic proteins. The FMRP mRNA complexes are then exported to the cytoplasm, from where they are trafficked to dendrites near neuronal synapses, figure 5.26. FRMP is a translation regulator. As synaptic junctions, FMRP suppresses protein synthesis from the bound mRNAs in response to signaling through group 1 metabotropic glutamate receptors, MGLUR. Thus, a reduction in FMRP and FXS results in increased translation of the bound mRNAs at synapses. This leads to an imbalance. Single gene disorders with non-classic inheritance 177. FMRP, axon, dendrite, ribosomes, axonal proteins. FMRP, mRNA, complex, ribosomes, axonal proteins. Figure 5.2.6, a model for the action of familial mental retardation protein FMRP in neurons. Modified from HENP, Warren Saint, New Insights into Fragile X Syndrome, From Molecules to Neurobehavior, Trends Biochem SCI. In the production of proteins at the synapses resulting in loss of synaptic plasticity, the ability of synapses to change and adapt in response to specific signals. Synaptic plasticity is essential for learning and memory. Although demonstration of an abnormal karyotype led to the identification of this disorder, polymerase chain reaction PCR-based detection of the repeats is now the method of choice for diagnosis. Fragile X-associated tremor, ataxia syndrome, and Fragile X-associated primary ovarian failure. Although initially assumed to be innocuous, CGG permutations in the FMR1 gene can cause 2 disorders that are phenotypically different from FXS and occur through a distinct mechanism involving a toxic gain of function. A decade after the discovery that CGG repeat expansions cause FXS, it became clear that approximately 20% of females carrying the premutation carrier females have premature ovarian failure before the age of 40 years. This condition is called Fragile X-associated primary ovarian failure. Affected women have menstrual irregularities and decreased fertility. FSH levels are elevated and anti-Müllerian hormone levels are decreased, both markers of declining ovarian function. They develop menopause approximately 5 years earlier than controls. Approximately 50% of premutation-carrying males, transmitting males, exhibit a progressive neurodegenerative syndrome starting in their 6th decade. This syndrome, referred to as Fragile X-associated tremor ataxia, is characterized by intention tremors and cerebellar ataxia and may progress to Parkinsonism. How do permutations cause disease? In these patients, the FMR1 gene, instead of being methylated in silence, continues to be transcribed. CGG containing FMR1 mRNAs, so formed, are toxic. They recruit RNA binding proteins and impair their function by sequestration from their normal locales. The expanded FMR1 mRNA and the sequestered RNA binding proteins aggregate in the nucleus and form intranuclear inclusions in both the central and the peripheral nervous systems. As in FXS, males are affected much more frequently and more severely than female permutation carriers. The pathogenesis of Fragile X-associated primary ovarian insufficiency is less well understood. Aggregates containing FMR1 mRNA have been detected in granulosa cells and ovarian stromal cells. Perhaps these aggregates cause premature death of ovarian follicles. Key concepts: Fragile X syndrome, FXS. Pathologic amplification of trinucleotide repeats causes loss of function, FXS, or gain of function, Huntington disease mutations. Most such mutations produce neurodegenerative disorders. FXS results from loss of FMR1 gene function and is characterized by severe intellectual disability and a variety of neuropsychiatric conditions such as autism spectrum disorders. In the normal population, there are about 29 to 55 CGG repeats in the FMR1 gene. The genomes of carrier males and females contain permutations with 55 to 200 CGG repeats. That can expand to 4,000 repeats full mutations during oogenesis. When full mutations are transmitted to progeny, FXS occurs. Carriers of permutations develop Fragile X-associated tremor, ataxia, and Fragile X-associated primary ovarian failure due to toxic gain of function by the abnormal FMR1 mRNA. Mutations in mitochondrial genes, Leber hereditary optic neuropathy. The vast majority of genes are located on chromosomes in the cell nucleus and are inherited in classic Mendelian fashion. Several mitochondrial genes exist, however, that are inherited in quite a different manner. A feature unique to mtDNA is maternal inheritance. This peculiarity exists because ova contain numerous mitochondria within their abundant cytoplasm, whereas spermatozoa contain few, if any. Hence, the mtDNA complement of the zygote is derived entirely from the ovum. Thus, mothers transmit mtDNA to all their offspring, male and female, daughters, but not sons, transmit the DNA further to their progeny. Figure 527. Several other features apply to mitochondrial inheritance. Human mtDNA contains 37 genes, of which 22 are transcribed into transfer RNAs and 2 into ribosomal RNAs. The remaining 13 genes encode subunits of the respiratory chain enzymes. Because mtDNA encodes enzymes involved in oxidative phosphorylation, mutations affecting these genes exert their deleterious effects primarily on the organs most dependent on oxidative phosphorylation, such as the central nervous system, skeletal muscle, cardiac muscle, liver, and kidneys. Each mitochondrion contains thousands of copies of mtDNAs, and typically deleterious mutations of mtDNA affect some, but not all, of these copies. Thus, tissues, and indeed, individuals may harbor both wild-type and mutant mtDNA, a situation called heteroplasmy. A minimum number of mutant mtDNAs must be present in a cell, or 178, Chapter 5, Genetic Disorders. Figure 5. Pedigree of Leber hereditary optic neuropathy, a disorder caused by mutation in mitochondrial DNA. Note that all progeny of an affected male shaded squares are normal, but all children, male and female, of the affected female shaded circles manifest disease to a variable degree, as discussed in the text. Tissue before oxidative dysfunction gives rise to disease. This is called the threshold effect. Not surprisingly, the threshold is reached most easily in the metabolically active tissues listed earlier. During cell division, mitochondria and their contained DNA are randomly distributed to the daughter cells. Thus, when a cell containing normal and mutant mtDNA divides, the proportion of the normal and mutant mtDNA in daughter cells is extremely variable. Therefore, the expression of disorders resulting from mutations in mtDNA is quite variable. Diseases associated with mitochondrial inheritance are rare, and, as mentioned earlier, many of them affect the neuromuscular system. Leber hereditary optic neuropathy is a prototype of this type of disorder. It is a neurodegenerative disease that manifests as progressive bilateral loss of central vision. Visual impairment is first noted between ages 15 and 35, leading eventually to blindness. Cardiac conduction defects and minor neurologic manifestations have also been observed in some families. Genomic imprinting. We all inherit 2 copies of each autosomal gene, carried on homologous maternal and paternal chromosomes. In the past, it had been assumed that there is no functional difference between the alleles derived from the mother or the father. Studies have now provided definite evidence that, at least with respect to some genes, important functional differences exist between the paternal allele and the maternal allele. These differences result from an epigenetic process called imprinting. In most cases, imprinting selectively inactivates either the maternal or the paternal allele. Thus, maternal imprinting refers to transcriptional silencing of the maternal allele, whereas paternal imprinting implies that the paternal allele is inactivated. Imprinting occurs in the ovum or the sperm before fertilization and then is stably transmitted to all somatic cells through mitosis. As with other instances of epigenetic regulation, imprinting is associated with differential patterns of DNA methylation at CG nucleotides. Other mechanisms include histone H4 deacetylation and methylation, Chapter 1. Regardless of the mechanism, it is believed that such marking of paternal and maternal chromosomes occurs during gametogenesis, and thus it seems that from the moment of conception, some chromosomes remember where they came from. The exact number of imprinted genes is not known. Estimates range from 200 to 600, although imprinted genes may occur in isolation. More commonly, they are found in groups that are regulated by common cis-acting elements called imprinting control regions. Genomic imprinting is best illustrated by two uncommon genetic disorders, Prader-Willi syndrome and Angelman syndrome, which were originally believed to be unrelated until the genetic lesions responsible for them were mapped to the same location. They are described next. Prader-Willi syndrome and Angelman syndrome. Prader-Willi syndrome is characterized by intellectual disability, short stature, hypotonia, profound hyperphagia, obesity, small hands and feet, and hypogonadism. In 65% to 70% of cases, an interstitial deletion of band Q12 in the long arm of chromosome 15, del 15 Q11 to Q13, can be detected. In most cases, the breakpoints are the same, causing a 5MB deletion. It is striking that in all cases, the deletion affects the paternally derived chromosome 15. In contrast to Prader-Willi syndrome, patients with the phenotypically distinct Angelman syndrome are born with a deletion of the same chromosomal region derived from their mothers. Patients with Angelman syndrome also have intellectual disability, but in addition, they present with microcephaly, ataxic gait, seizures, and inappropriate laughter. Because of their laughter and ataxia, they have been referred to as happy puppets. A comparison of these two syndromes clearly demonstrates the parent-origin effects on gene function. The molecular basis of these two syndromes lies in the process of genomic imprinting, figure 5.28. Three mechanisms are involved. Deletions. It is known that a gene or set of genes on maternal chromosome 15q12 is imprinted and hence silenced, and thus the only functional alleles are provided by the paternal chromosome. When these are lost as a result of a deletion, the person develops Prader-Willi syndrome. Conversely, a distinct gene that also maps to the same region of chromosome 15 is imprinted on the paternal chromosome. Only the maternally derived allele of this gene is normally active. Deletion of this maternal. Disorders with non-classic inheritance 179. Maternal-paternal. MEP, imprinted Prader-Willi, genes. Active Angelman gene, UB3A, active Prader-Willi, genes, imprinted Angelman, gene UB3A, MP, deletion in maternal. Chromosome, deletion in paternal. Chromosome MP, site of deletion. Active Prader-Willi, genes. Inactive UB3A gene, imprinted Prader-Willi, genes. Active UB3A gene. Site of deletion. Angelman syndrome, Prader-Willi syndrome. Figure 5.28 diagrammatic representation of Prader-Willi and Angelman syndromes. Gene on chromosome 15 gives rise to the Angelman syndrome. Deletions account for about 70% cases. Uniparental disomy. Molecular studies of cytogenetically normal individuals with Prader-Willi syndrome, i.e., those without the deletion, have revealed that they have two maternal copies of chromosome 15. Inheritance of both chromosomes of a pair from one parent is called uniparental disomy. The net effect is the same, i.e., the person does not have a functional set of genes from the non-imprinted paternal chromosomes 15. Angelman syndrome, as might be expected, can also result from uniparental disomy of paternal chromosome 15. This is the second most common mechanism, responsible for 20% to 25% cases. Defective imprinting. In a small minority of patients, 1% to 4%, there is an imprinting defect. In some patients with Prader-Willi syndrome, the paternal chromosome carries the maternal imprint. And conversely, in Angelman syndrome, the maternal chromosome carries the paternal imprint. Hence, there are no functional alleles. The genetic basis of these two imprinting disorders is now being unraveled. In Angelman syndrome, the affected gene is a ubiquitin ligase that is involved in catalyzing the transfer of activated ubiquitin to target protein substrates. The gene, called UB3A, maps within the 15q12 region, is imprinted on the paternal chromosome, and is expressed from the maternal allele primarily in specific regions of the brain. absence of UBE3A inhibits synapse formation and synaptic plasticity. The imprinting is tissue-specific, in that UBE3A is expressed from both alleles in most tissues. In contrast to Angelman syndrome, no single gene has been implicated in Prader-Willi syndrome. Instead, a series of genes located in the 15q11.2, q13 interval, which are imprinted on the maternal chromosome and expressed from the paternal chromosome, are believed to be involved. These include the SNORP family of genes that encode small nucleolar RNAs. These RNAs are non-coding molecules that are involved in post-transcriptional modifications of ribosomal RNAs and other small nuclear RNAs. Loss of SNORP functions is believed to contribute to Prader-Willi syndrome, but the precise mechanisms are unclear. Molecular diagnosis of these syndromes is based on assessment of methylation status of marker genes in fish. The importance of imprinting is not restricted to rare chromosomal disorders. Parent of origin effects have been identified in a variety of inherited diseases, such as Huntington disease and myotonic dystrophy, and in tumorigenesis. Key concepts: genomic imprinting. Imprinting involves transcriptional silencing of the paternal or maternal copies of certain genes during gametogenesis. For such genes, only one functional copy exists in the individual. Loss of the functional not imprinted allele by deletion gives rise to diseases. Single gene. Chapter 5. Genetic disorders. In Prader-Willi syndrome, there is deletion of band Q12 on the long arm of paternal chromosome 15. Genes in this region of maternal chromosome 15 are imprinted, so there is complete loss of their functions. Patients have intellectual disability, short stature, hypotonia, hyperphagia, small hands and feet, and hypogonadism. In Angelman syndrome, there is deletion of the same region from the maternal chromosome, and genes on the corresponding region of paternal chromosome 15 are imprinted. These patients have intellectual disability, ataxia, seizures, and inappropriate laughter. 180. Gonadal mosaicism. It was mentioned earlier that with every autosomal dominant disorder, some patients do not have affected parents. In such patients, the disorder results from a new mutation in the egg or the sperm from which they were derived. As such, their siblings are neither affected nor at increased risk for development of the disease. This is not always the case, however. In some autosomal dominant disorders, exemplified by osteogenesis imperfecta, phenotypically normal parents have more than one affected child. This clearly violates the laws of Mendelian inheritance. Studies indicate that gonadal mosaicism may be responsible for such unusual pedigrees. Such mosaicism results from a mutation that occurs post-zygotically during early embryonic development. If the mutation affects only cells destined to form the gonads, the gametes carry the mutation, but the somatic cells of the individual are completely normal. A phenotypically normal parent who has gonadal mosaicism can transmit the disease-causing mutation to the offspring through their mutated gametes. Because the progenitor cells of the gametes carry the mutation, there is a possibility that more than one child of such a parent would be affected. Obviously, the likelihood of such an occurrence depends on the proportion of germ cells carrying the mutation. Molecular genetic diagnosis. The field of molecular genetic diagnostics emerged in the latter half of the 20th century in the form of labor-intensive, low-throughput methods such as conventional karyotyping for diagnosis of cytogenetic disorders, e.g. Down syndrome, and DNA-based assays such as southern blotting for the diagnosis of Huntington disease. Over time, a steady stream of technologic breakthroughs has led to ever-increasing capabilities, particularly in the area of nucleic acid sequencing. These advances began with the development of Sanger DNA sequencing in 1977 and PCR in 1983 and accelerated rapidly with the advent of high throughput, massively parallel sequencing strategies, often lumped together under the rubric next-generation sequencing (INGS) in the late 1990s. NGS has continued to improve in terms of speed and cost, and it is now possible to sequence an entire genome for less than $1,000 in a few days. As a result, nucleic acid-based testing is taking a central role in the diagnosis and management of many diseases, and we currently are in a transition period during which many simple, single-gene tests using old technologies are being slowly but surely supplanted by more comprehensive NGS-based approaches. That said, certain types of genetic lesions are difficult to detect by NGS, and for the foreseeable future, older technologies will continue to have an important role in testing for genetic diseases. An exhaustive discussion of molecular diagnostics is beyond the scope of this book. Here, we highlight some of the most useful and widely deployed approaches. When considering molecular genetic testing, it is important to remember that genetic markers can be either constitutional, i.e., present in every cell of the affected person, as with the CFTR mutation in a patient with cystic fibrosis, or somatic, i.e., restricted to specific tissue types or lesions, as with mutations in RAS in a variety of human cancers. Similarly, in suspected infections, nucleic acids that are specific to the infectious agent may be confined to particular cells or body sites. These considerations determine the nature of the sample used for the assay, e.g., peripheral blood cells, tumor tissue, nasopharyngeal swab. Diagnostic methods and indications for testing. There are a dizzying number of techniques and indications for performing molecular genetic diagnostic tests on patient specimens. The burden of choice is often problematic, both for molecular pathologists who design tests and for clinicians who need to choose the optimal test for their patients. Laboratory considerations. Pathologists developing tests focus on the sensitivity, specificity, accuracy, and reproducibility of different methods, as well as practical factors like cost, labor, reliability, and turnaround time. To choose the most appropriate diagnostic technique, one must have a thorough knowledge of the spectrum of genetic anomalies that are responsible for the disease in the patient population under study. Disease-causing genetic anomalies range in size from single-base substitutions to gains or losses of entire chromosomes, and often vary widely in frequency among ethnic groups. Proper test design requires careful consideration of these factors. For example, standard cystic fibrosis testing for pathogenic mutations in CFTR has a sensitivity of 94% in Ashkenazi Jews, but identifies less than 50% of affected patients in Asian populations. In cases with negative standard test results and a high clinical suspicion, further testing is indicated. However, even with extensive sequencing of CFTR, no pathogenic mutations are identified in approximately 10% of patients with classic cystic fibrosis. This is likely because NGS assays miss certain genetic lesions, e.g., kilobase scale deletions and gene rearrangements, that are better detected with other methods. Issues like this arise frequently in genetic testing and close communication between primary care clinicians, medical genetics specialists, and diagnosticians is required to select the optimal test strategy in difficult cases. Indications for analysis of inherited genetic alterations. Although it is not unusual for inherited genetic disorders to present in adulthood, most testing is performed during the prenatal or postnatal childhood periods. Mendelian disorders caused by mutations in specific genes number in the thousands, and definitive diagnosis of most of them is possible by DNA sequencing tests. Genetic diagnosis 181. Other inherited disorders are the result of chromosomal aberrations that usually present prenatally or at birth. Prenatal testing should be offered for all fetuses at risk for a cytogenetic abnormality. Possible indications include the following: advanced maternal age, a parent known to carry a balanced chromosomal rearrangement, which greatly increases the frequency of abnormal chromosome segregation during meiosis and the risk of aneuploidy in the fertilized ovum. Fetal anomalies observed on ultrasound. Routine maternal blood screening indicating an increased risk of Down syndrome, trisomy 21, or another trisomy. Prenatal testing may also be considered for fetuses at known risk for Mendelian disorders, e.g., cystic fibrosis, spinal muscular atrophy, based on family history. At present, it is usually performed on cells obtained by amniocentesis, chorionic villus biopsy, or umbilical cord blood. However, as much as 10% of the free DNA in a pregnant mother's blood is of fetal origin, and new sequencing technologies have opened the door to an era of non-invasive prenatal diagnostics utilizing this source of DNA. Following birth, testing is ideally done as soon as the possibility of constitutional genetic disease arises. It is most commonly performed on peripheral blood DNA and is targeted based on clinical suspicion. In newborns or children, indications are as follows: multiple congenital anomalies, suspicion of a metabolic syndrome, unexplained intellectual disability and/or developmental delay, suspected aneuploidy, e.g., features of Down syndrome or other syndromic chromosomal abnormality, e.g., deletions, inversions, suspected monogenic disease, whether previously described in the family or new. In older patients, testing logically becomes more focused toward genetic diseases that manifest at later stages of life. Again, the possibilities are vast, but the more common indications include the following: inherited cancer syndromes triggered by either family history or an unusual cancer presentation, such as multiple cancer types or unusually young age at diagnosis. Atypically mild monogenic disease, e.g., attenuated cystic fibrosis, Neurodegenerative disorders, e.g., familial Alzheimer's disease, Huntington disease. Indications for analysis of acquired genetic alterations. In this era of molecularly targeted therapies, it is increasingly important to identify nucleic acid sequences or aberrations that are specific for acquired diseases, e.g., cancer and infectious disease. The technical approaches are the same as those used for germline Mendelian disorders. Common indications include the following: diagnosis and management of cancer. Chapter 7. Detection of tumor-specific mutations and cytogenetic alterations that are the hallmarks of specific tumors, e.g. BCR-ABL fusion genes and chronic myeloid leukemia, CML molecular. Determination of clonality as an indicator of a neoplastic condition. Identification of specific genetic alterations that can direct therapeutic choices, e.g. amplification of HER2, official gene name ERBB2, in breast cancer, or mutation of EGFR, official gene name ERB1, in lung cancer. Determination of treatment efficacy, e.g., minimal residual disease detection in patients with CML by quantitative PCR for BCR-ABL. Detection of drug-resistant secondary mutations in malignancies treated with therapies that target specific proteins, e.g., mutated EGFR. Diagnosis and management of infectious disease, Chapter 8. Detection of microorganism-specific genetic material for definitive diagnosis, e.g., human immunodeficiency virus, HIV, mycobacteria, human papillomavirus, herpes virus in central nervous system. Identification of genetic alterations in the genomes of microbes that are associated with drug resistance. Determination of treatment efficacy, e.g., assessment of viral loads in HIV, Epstein-Barr virus, and hepatitis C virus infection. PCR and detection of DNA sequence alterations. PCR analysis, which involves synthesis of relatively short DNA fragments from a DNA template, has been a mainstay of molecular diagnostics for the last few decades. By using appropriate heat-stable DNA polymerases and thermal cycling, the target DNA, usually less than 1000 BP lying between designed primer sites, is exponentially amplified from as little as one original copy, greatly simplifying secondary sequence analysis. Many options exist for subsequent analysis, each with different strengths and weaknesses. A few of the more common options are described below. Sanger sequencing. A single PCR product is mixed with a DNA polymerase, a specific primer, nucleotides, and 4 dead-end di-deoxy terminator nucleotides, A, T, G, and C, labeled with different fluorescent tags. The ensuing reaction produces a ladder of DNA molecules of all possible lengths, each labeled with a tag corresponding to the base at which the reaction stopped due to incorporation of a terminator nucleotide. After size separation by electrophoresis, the sequence is read and compared with the normal sequence to detect mutations. Next-generation sequencing, NGS. PCR using primers for many different genomic regions is performed simultaneously, and the resulting mixture of PCR products enriched for regions of interest are subjected to NGS, described in more detail later. NGS is more sensitive than Sanger sequencing, as it can reliably identify the presence of mutations in only a small percentage of the individual sequencing reads. This situation is encountered frequently in cancers, either because the mutation in question is only present in a small fraction of tumor cells, or because the tumor cells are heavily contaminated with genetically normal stromal cells. Single-base primer extension. This is a useful approach for identifying mutations at a specific nucleotide position, e.g., an oncogenic mutation in codon 600 of the BRAF gene. A primer is added to the PCR product that 182, Chapter 5, Genetic Disorders, hybridizes one base upstream of the target. Differently colored terminator fluorescent nucleotides are added corresponding to normal and variant bases, and a single base polymerase extension is performed. The relative amounts of normal variant fluorescence are then detected, figure 5.29. This technique is very sensitive, but has the obvious disadvantage of only producing 1 BP of sequence data. Restriction fragment length analysis. This simple approach takes advantage of the digestion of DNA with endonucleases known as restriction enzymes that recognize and cut DNA at specific sequences. If the specific mutation is known to affect a restriction site, the amplified PCR product may be digested, and the normal and mutant PCR products will yield fragments of different sizes that are easily distinguished. Needless to say, this approach is considerably less comprehensive than direct sequencing, but remains useful for molecular diagnosis when the causal mutation always occurs at an invariant nucleotide position. Point mutation, DNA GA primer, CT, TC, fluorescently labeled nucleotides, normal permutation, full mutation, ACOR iota recognition sites, probe. Normal permutation full. Mutation, southern blot, CGG repeat, region, normal permutation full, mutation, PCR. Figure 5.30, diagnostic application of polymerase chain reaction PCR and southern blot analysis in Fragile X syndrome with PCR. The differences in the size of CGG repeats between normal and premutation give rise to products of different sizes and mobility. With a full mutation, the region between the primers is too large to be amplified by conventional PCR. In southern blot analysis, the DNA is cut by enzymes that flank the CGG repeat region and is then probed with complementary DNA that binds to the affected part of the gene. A single small band is seen in normal males, a band of higher molecular weight is seen in males with premutation, and a very large, usually diffuse band is seen in males with the full mutation. PCR primers. Coding sequence of FMR1 gene, wild-type DNA. Figure 5.29 single-base extension analysis of a polymerase chain reaction product, using a primer to interrogate a single-base position. Nucleotides complementary to the mutant and wild-type bases at the queried position are labeled with different fluorophores, such that incorporation yields fluorescent signals of varying intensity based on the ratio of mutant to wild-type DNA present. Amplicon length analysis. Mutations that affect the length of DNA, e.g. deletions or expansions, can be easily detected by PCR. As discussed earlier, several diseases, such as FXS, are associated with alterations in trinucleotide repeats. Figure 5.30 reveals how PCR analysis can be used to detect this mutation. Two primers that flank the region containing the trinucleotide repeats at the 5' end of the FMR1 gene are used to amplify the intervening sequences. Because there are large differences in the number of repeats, the size of the PCR products obtained from the DNA of normal individuals or those with a premutation is quite different and can be easily distinguished by gel electrophoresis. An important caveat is that if the mixed wild type and mutant DNA, trinucleotide expansion is so large that it cannot be amplified by convention PCR, southern blot analysis of genomic DNA may have to be performed. As discussed earlier, large expansions are not uncommon in FXS, real-time PCR. A variety of PCR-based technologies that use fluorophore indicators can detect and quantify the presence of particular nucleic acid sequences in real time, i.e. during the exponential phase of DNA amplification rather than post-PCR. It is most often used to monitor the frequency of cancer cells bearing characteristic genetic lesions in the blood or in tissues, e.g. the level of BCR-ABL fusion gene sequences in patients with CML, or the infectious load of certain viruses, e.g. HIV, Epstein-Barr virus. Molecular analysis of genomic alterations. A significant number of genetic lesions involve large deletions, duplications, or more complex rearrangements that are not readily amenable to detection using PCR or DNA sequencing approaches. Such genomic scale alterations can be studied using a variety of hybridization-based techniques. Fluorescence in situ hybridization FISH uses DNA probes that recognize sequences specific to particular chromosomal regions. To perform FISH, large fragments of cloned genomic DNA spanning up to 200 kilobits are labeled with fluorescent dyes and applied to genetic diagnosis 183. Metaphase chromosome preparations or interphase nuclei that are pre-treated so as to melt the genomic DNA. The probe hybridizes to its homologous genomic sequence and thus labels a specific chromosomal region that can be visualized under a fluorescent microscope. FISH can be performed on prenatal samples, peripheral blood cells, touch preparations from cancer biopsies, and even fixed archival tissue sections. It is used to detect numeric abnormalities of chromosomes aneuploidy, see Figure 519, subtle microdeletions, see Figure 5.21, and complex translocations that are not demonstrable by routine karyotyping. And gene amplification, e.g. HER2 in breast cancer, or NYC amplification in neuroblastomas. It is also used in certain circumstances when rapid diagnosis is essential, e.g. when deciding to treat a patient with suspected acute promyelocytic leukemia with retinoic acid, which is effective only when a particular chromosomal translocation involving the retinoic acid receptor gene is present in the tumor cells, Chapter 13. Cytogenomic array technology FISH requires prior knowledge of the one or few specific chromosomal regions suspected of being altered in the test sample. However, genomic abnormalities can also be detected without prior knowledge by using microarray technology to perform a global genomic survey. First-generation platforms were designed for comparative genomic hybridization CGH, while newer platforms incorporate single nucleotide polymorphism SNP genotyping approaches. offering multiple benefits. Array-based comparative genomic hybridization. In array CGH, the test DNA and a reference normal DNA are labeled with two different fluorescent dyes. The samples are then mixed and hybridized to an array spotted with DNA probes that span the human genome at regularly spaced intervals. At each chromosomal probe location, the binding of the labeled DNA from the two samples is compared. If the two samples are equal, i.e. the test sample is diploid, all spots on the array will fluoresce yellow due to equal admixture of green and red dyes. In contrast, if the test sample has a deletion or duplication, the probe spots corresponding to it will show skewing toward red or green depending on gain or loss of material, allowing highly accurate determination of even focal copy number, gain or loss, anywhere across the genome. Single nucleotide polymorphism genotyping arrays. Newer types of genomic arrays are based on a similar concept, but some or all of the probes are designed to identify SNP sites genome-wide, which provides a number of advantages. As discussed in Chapter 1, SNPs are the most common type of DNA polymorphism, occurring on average approximately every 1,000 nucleotides throughout the genome. They serve both as a physical landmark within the genome and as a genetic marker whose transmission can be followed from parent to child. There are several testing platforms using different methodologies that allow SNPs to be analyzed genome-wide on arrays. Details of these methods are beyond the scope of this discussion. Like CGH probes, these methods involving SNPs can be used to make copy number variation CNV calls. But by discriminating between SNP alleles at each particular location, they also provide zygosity data, figure 5.31. The current generation of SNP arrays is quite comprehensive, with the largest containing greater than 4 million SNP probes. 2. Genomic DNA 1, 0. Copy number 1, 2, 1 AA 0.8 SNP array hybridization, zygosity, AB, BB, 0.64, 02, 0. 12. P12 copy number variation, one analysis of copyotide polymorphism, SNP. Genomic DNA is labeled and hybridized to an array containing potentially millions of probe spots. Copy number is determined by overall intensity, and genotype is determined by allelic ratio. The example shown is the P arm of chromosome 12 in a patient with pediatric leukemia. The normal areas green show neutral diploid DNA content, and the zygosity plot shows the expected ratio of AA, AB, and BB SNP genotypes. The anomalous area red shows decreased overall intensity, and the zygosity plot shows absence of the mixed AB genotype, indicating a full heterozygous deletion. Modified from Paulson K. et al. Genetic landscape of high hyperdiploid childhood acute lymphoblastic leukemia, Procnatal ACAS Psi USA 721, 21724, 2010. 184, Chapter 5, Genetic Disorders. In the clinical laboratory, SNP arrays are routinely used to uncover copy number abnormalities in pediatric patients when the karyotype is normal, but a structural chromosomal abnormality is still suspected. Common indications include congenital abnormalities, dysmorphic features, developmental delay, and autism. Notably, unlike CDH arrays, SNP arrays can identify loss of heterozygosity. This capability is important in the diagnosis of disorders caused by uniparental disomy, e.g., Prader-Willi syndrome and Angelman syndrome, where despite diploid genomic copy number, the SNP calls in the affected chromosomal regions showed reduction to homozygosity. SNP data can also help uncover other anomalies, such as mosaicism, which produces complex but distinctive skewing of zygosity plots. Polymorphic markers and molecular diagnosis. Clinical detection of disease-specific mutations is possible only if the gene responsible for the disorder is known and its sequence has been identified. If the exact nature of the genetic aberration is not known, or if testing for the primary defect is technically challenging or unfeasible, diagnostic laboratories can take advantage of the phenomenon of linkage. In humans, two DNA loci, even 100,000 BP apart on the same chromosome, are almost certain to co-segregate during meiosis due to the extremely low chance of a crossover event happening between them. Thus, the closer two loci are, the higher the probability that they will travel together in family pedigrees. In the event of a challenging or unknown pathogenic allele, a diagnostic laboratory can instead choose as a surrogate approach to examine nearby marker loci in the context of the family pedigree. The two types of genetic polymorphisms most useful for linkage analysis are SNPs described earlier and repeat-length polymorphisms known as mini-satellite and microsatellite repeats. Human DNA contains short repetitive sequences of DNA, giving rise to what are called repeat-length polymorphisms. These polymorphisms are often subdivided on the basis of their length into microsatellite repeats and mini-satellite repeats. Microsatellites are usually less than 1 kilobit and are characterized by a repeat size of 2 to 6 BP. Mini-satellite repeats, by comparison, are larger 1 to 3 kilobits, and the repeat motif is usually 15 to 70 BP. The number of repeats, both in microsatellites and in mini-satellites, is extremely variable within a given population, and hence, these stretches of DNA can be used to establish genetic identity for linkage analysis. Different microsatellites and mini-satellites are readily distinguishable on the basis of size, which can be measured by doing PCR amplification using primers that flank the repeat region. Figure 5.3.2 depicts the application of microsatellite linkage analysis to the PKD1 gene, historically very difficult to sequence for the familial diagnosis of adult polycystic kidney disease. It can be seen that the longest microsatellite allele is linked in the family to the disease allele and can be used to track its transmission. Assays to detect genetic polymorphisms are also important in many other areas of medicine, including in the determination of relatedness and identity in transplantation, cancer genetics, paternity testing, and forensic medicine. Since microsatellite markers are scattered throughout the human genome and have such a high level of polymorphism, they are ideal for differentiating between two individuals and to follow transmission of the marker from parent to child. Assays employing PCR amplification of informative microsatellite markers are now routinely used for determining paternity and for criminal investigations. PCR can be performed even with highly degraded biologic samples, allowing DNA testing to play a central role in forensic identifications. The same assays are used in patients following allogeneic hematopoietic stem cell transplant to detect and quantify chimerism, which is evaluated by assessing the relative amounts of donor-specific and host-specific microsatellite markers in the patient's blood cells. Epigenetic alterations. Epigenetics is defined as the study of heritable chemical modification of DNA or chromatin that does not alter the protein encoding DNA sequence itself. Examples of such modifications include methylation of DNA and methylation and acetylation of histones. Chapter 1. Our understanding of the importance of these types of molecular alterations is rapidly growing, and it is clear that epigenetic modifications are critical for normal human development, including the regulation of tissue-specific gene expression, X chromosome inactivation, and imprinting, as well as for understanding the cellular perturbations in aging and cancer. Allele, allele B, allele C, A, ketxexexesa, saktecha sakechi, tetsachi. PCR primers, PKD gene, CAB, AC, AB, BA, before Christ AB, CB before Christ B, BB. Figure 5. DNA polymorphisms resulting from a variable number of CA repeats. The three alleles A produce polymerase chain reaction PCR products of different sizes B, thus identifying their origins from specific chromosomes. In the example depicted, allele C is linked to a mutation responsible for autosomal dominant polycystic kidney disease, PKD. Application of this to detect progeny carrying the disease-related gene, red symbols, is illustrated in one hypothetical pedigree. Male squares, female circles. Molecular genetic diagnosis 185. Gene expression frequently correlates negatively with the level of methylation of DNA, particularly of cytosine residues in CG dinucleotide-rich promoter regions known as CPG islands. As discussed earlier in the section on genomic imprinting, increased methylation of CPG islands is associated with decreased gene expression and is accompanied by concomitant alterations of histone methylation and acetylation. Diagnosis of an increasing number of diseases involves the analysis of promoter methylation, for example, FXS, in which hypermethylation results in FMR1 silencing. Methylation analysis is also essential in the diagnosis of Prader-Willi syndrome and Angelman syndrome. Special techniques are needed to detect DNA methylation. One approach is to treat genomic DNA with sodium bisulfite, a chemical that converts unmethylated cytosine to uracil, which acts like thymine in downstream reactions. Methylated cytosines are protected from modification and remain unchanged. After treatment, it is then straightforward to discriminate the unmethylated modified DNA from the methylated unmodified DNA by DNA sequencing. RNA analysis. Because DNA exerts its effects on the cell through RNA expression and mature mRNA contains the coding sequences of all expressed genes. RNA can in principle substitute for DNA in a wide range of diagnostic applications. From a practical standpoint, DNA-based diagnosis is usually preferred, since DNA is much more stable. Nonetheless, RNA analysis is critical in several areas of molecular diagnostics. The most important applications are the detection and quantification of RNA viruses such as HIV and hepatitis C virus, but increasingly RNA analysis is also being used to evaluate cancer. mRNA expression profiling, described for breast cancer in Chapter 23, has become an important tool for molecular stratification of certain tumors. In some instances, cancer cells bearing particular chromosomal translocations are detected with greater sensitivity by analyzing mRNA, e.g., the BCR-ABL fusion transcript in CML. The principal reason for this is that most translocations occur in scattered locations within particular introns, which can be very large, complicating detection by PCR amplification of DNA. Since introns are removed by splicing during the formation of mRNA, PCR analysis is possible if RNA is first converted to complementary DNA cDNA by reverse transcriptase. Real-time PCR performed on cDNA is the method of choice for monitoring residual disease in patients with CML and certain other hematologic malignancies, Chapter 13. Next-generation sequencing, NGS. Next-generation sequencing is a term used to describe several newer DNA sequencing technologies that produce very large amounts of sequence data in a massively parallel manner. These technologies have revolutionized biomedical research and are increasingly impacting molecular diagnostics. The factors propelling rapid adoption of NGS are price and performance. NGS allows us to perform previously impossible analyses at extremely low relative cost. One feature that makes NGS far more clinically applicable than Sanger sequencing is its input sample requirement. Whereas Sanger sequencing requires a single homogeneous template DNA, usually a specific PCR product, NGS has no such requirement. Any DNA from almost any source can be used. Because Sanger sequencing essentially provides an average result for a particular DNA region in a sample, samples with substantial sequence heterogeneity among input DNA molecules produce uninterpretable results. NGS, by contrast, is well suited to analysis of heterogeneous DNA samples due to the application of three common basic principles, Figure 5.33, spatial separation. At the beginning of the procedure, individual DNA molecules are physically isolated from each other in space. The specifics of this process are platform dependent. Local amplification. After separation, the individual DNA molecules are amplified in place using a limited number of PCR cycles. Amplification is necessary so that sufficient signal can be generated to ensure detection and accuracy. Parallel sequencing. The amplified DNA molecules are then simultaneously sequenced by the addition of polymerases and other reagents, with each spatially separated and amplified original molecule yielding a read corresponding to its sequence. Sequence reads from NGS instruments are generally short, less than 500 BP. Bioinformatics. Each NGS analysis generates a staggering amount of sequence data. For high-throughput sequencing instruments, this amounts to 400 billion BP or more of sequence per day, enough to produce a high-quality sequence spanning an entire human genome. The downstream analysis necessary to make sense of these enormous data sets is sufficiently complex that specialized training in bioinformatics is needed to ensure its proper interpretation. Bioinformatics. Computational pipelines vary widely across applications and sample types, and a detailed discussion is beyond the scope of this text. However, it is worth describing the basic steps necessary to process this type of data. Alignment. Alignment is the process by which the sequencing reads from a sample are mapped onto a reference genome, allowing them to be viewed and interpreted in context. Variant calling. This process involves a systematic comparison of all of the sequence data from a sample with the reference sequence. The more reads that cover a particular location sequencing depth, the more likely that a variant will be detected if present. If a locus shows sufficient evidence of a difference from the reference sequence, a variant call is made. Variant annotation and interpretation. Called variants can be annotated with various features, e.g. gene name, coding change, and protein effect predictions, information from databases listing benign and pathogenic variants. Clinical information. Once completed by the clinical laboratory, the annotated data are ready for reporting. Because the pace of advances in genetics and genomics has far outpaced the knowledge of the typical clinician. An important part of this report is a brief description of the biological and clinical significance of each pathogenic variant, which can be numerous in the case of some cancers. Chapter 5. Genetic Disorders. AGCCTG, AAGCCTG, ACGTCGTCGTC, GGATCGGATC. Add all 4 nucleotides, labeled with different dyes wash, 4-color imaging cleave dye and terminating groups, wash repeat. BCATG Figure 5.3 Principle of Next Generation Sequencing. Several alternative approaches currently are available for next-generation sequencing, and one of the more commonly used platforms is illustrated. Short fragments of genomic DNA template 100 to 500 BP in length are immobilized on a solid phase platform such as a glass slide, using universal capture primers that are complementary to adapters that have previously been added to ends of the template fragments. The addition of fluorescently labeled complementary nucleotides, 1 per template DNA per cycle, occurs in a massively parallel fashion at millions of templates immobilized on the solid phase at the same time. A 4-color imaging camera captures the fluorescence emanating from each template location corresponding to the specific incorporated nucleotide, following which the fluorescent dye is cleaved and washed away, and the entire cycle is repeated. Powerful computational programs can decipher the images to generate sequences complementary to the template DNA at the end of one run. And these sequences are then mapped back to the reference genomic sequence to identify alterations. Reproduced with permission from Metzger-M. Sequencing Technologies, The Next Generation. NatRev Gen 113146, 2010, Nature Publishing Group. 186. Top. KTGT Bottom. CCCCC. Mutational signature calling. In addition to totaling up individual mutations, informatics algorithms have been developed that detect patterns of mutations that point to particular environmental exposures, e.g., ultraviolet light, or underlying defects in DNA repair. The latter has become clinically relevant with the discovery that cancers with acquired defects in mismatch repair genes that lead to microsatellite instability (MSI) are highly responsive to immune checkpoint inhibitors. Chapter 7. Based on this observation, many clinical laboratories are now calling the MSI status of cancers based on NGS results. Clinical applications of next-generation DNA sequencing. As discussed, any DNA sample can be analyzed by NGS. However, the DNA needs to first be prepared for sequencing by preparing a library of short DNA sequences that are enriched for genomic regions of interest, e.g., particular exons. There are several methods for NGS library preparation from genomic DNA, depending on the question and desired result. In the clinical laboratories, most applications of NGS are targeted toward constitutional genetic disease and cancer diagnostics, using a few different basic approaches. Targeted sequencing. Most current clinical laboratory NGS tests fall in this category. Targeted sequencing is focused on a carefully selected panel of genes, which maximizes sequencing depth while minimizing costs and the time and expense required for interpretation and clinical reporting. Sample preparation for targeted sequencing can be performed either by enriching for sequences of interest through a method called hybrid capture via custom, complementary probes, or by multiplex PCR. Single-gene NGS assays for inherited disorders are also becoming more common, and many laboratories that previously performed costly Sanger sequencing of multiple PCR products, i.e., to enable comprehensive CFTR sequencing, are now moving these assays to NGS platforms. Targeted NGS with gene panels is also now commonplace in evaluating individuals with genetic diseases such as cardiomyopathy and congenital deafness. In cancer testing, gene panels are widely used to perform detailed tumor profiling. Each tumor has a unique set of somatic mutations, and these assays aim to detect as many treatable or prognostic mutations as possible to offer individually tailored patient care. Increasingly, repeat testing at the time of disease recurrence is also being performed to understand mechanisms of drug resistance, which can serve to guide the selection of second-line therapies. Whole exome sequencing (WES). WES is a more expansive type of targeted sequencing in which hundreds of thousands of custom probes are used to enrich for the roughly 1.5% of the genome that consists of protein encoding exons. Prior to NGS, it is not routinely used in the evaluation of suspected germline disorders, but has. Readings 187 led to some wonderful success stories, allowing physicians to deliver answers and even therapies for children with orphan diseases who had suffered through prolonged and unsuccessful diagnostic odysseys. WES is also used in oncology to perform a very broad analysis, mostly in the research setting. Whole genome sequencing (WGS). WGS is the most comprehensive type of DNA analysis that can be performed on an individual. Current indications for use in medical genetics are mostly limited to cases where exome sequencing has failed to provide an answer, but the clinical suspicion of genetic disease remains high. For cancer applications, WGS is the only form of NGS that can detect novel structural rearrangements, e.g., insertions, deletions, translocations, that may be clinically relevant but relatively high costs. Slow turnaround times and significant informatics challenges still preclude its routine use in clinical practice. Future applications, because NGS can be used to detect genetic anomalies of essentially any size scale, from SNPs to very large rearrangements and even aneuploidy. Almost all of today's genetic diagnostic test modalities could in principle be supplanted by NGS. This includes RNA analysis because NGS-based analysis of the transcriptome RNA-seq is straightforward. As costs continue to drop, it is reasonable to expect NGS to occupy an increasingly prominent place in the diagnostics laboratory. Additionally, NGS has great promise for clinical applications in a number of other areas, including microbiome analysis, blood screening for early markers of diseases, e.g. cancer, and vastly more sensitive methods of gauging the response of cancers to therapy, i.e. using circulating free DNA released from tumor cells that is harvested from the blood. Continuing technologic advances may even extend the applications further. For example, third-generation single molecule or next-generation technologies are emerging that can rapidly sequence single molecules in parallel without the need for focal amplification, and these could soon have an impact in the clinical laboratory. Acknowledgement. The assistance of Jeremy Siegel, MD, PhD, Director, Division of Genomic and Molecular Pathology, University of Chicago, in the revision of the section on molecular diagnosis is greatly appreciated. Suggested readings: Molecular Basis of Single Gene Disorders, General Dietz HC, New Therapeutic Approaches to Mendelian Disorders, N. Engel J. Med 363-852, 2010. An excellent discussion of treatment of genetic disorders based on molecular pathogenesis. Disorders associated with defects in structural proteins. Cortini F. Villa C. Marinelli